So
Okay.
Taylor's going to do, does science suggest abiogenesis or intelligent design?
Yeah, I worked for a while on this presentation actually. I got it ready for another server and it's been a minute, but I'm gonna be doing a presentation on refraction coming up here. So I wanted to get this presentation out of the way because Rick's told me I can never watch it. And there's a lot of atheists and stuff who come in these servers, and I just want to like share like what I've encountered with the origin of life and the research that I've seen and encountered on it, just to help people get on track with the fact that these things do not come about on their own. There's a necessity for an intelligent designer and a creator. And yeah. Okay. Oops, let me go back. Okay, so I study biology and I have my undergrad in biology and I'm working on my graduate level degree in biology. Um and yeah, I've written papers on this before. So let me just give a little background. Biology is the study of life and living organisms. Cell biology is the study of cells in particular, which is what I study. And cells are the basic fundamental unit of life. And then a fun fact here is that cell is derived from the Latin term cellula, which means small room. Whenever they actually looked at them on the microscope, they looked like little tiny rooms. So that's where it got its name from. All right. And then there's different branches of biology. And cell biology is obviously what I'm preference towards, but a lot of things branch off of it, like genetics and anatomy, like looking at different parts of humans, anatomy and larger organisms. I look at like single-celled organisms. So I actually study like the organelles, but anyway, there's different branches of biology. And here's like the fundamentals of biology, the characteristics of life, and basically how we get to a cell in larger organisms. So we have like atoms, right, that make up they say everything pretty much. And then molecules, atoms come together to make molecules. Molecules will come together to make organelles. Organelles make up cells. Cells make up tissues. Tissues make up organ systems, which make up organisms, ecosystems, and the entire Earth, the entire biosphere, all life on Earth. And then there's characteristics of life. All right, here's the characteristics of something that's living. They're made of one or more cells. They have organization. Cells can grow and develop. They can reproduce, respond to stimuli, use energy, and maintain homeostasis. All right, here's a video I actually got on the left. Or a picture in the bottom left, and it came with a video, but I found a better video online. These are actually what single-celled organisms will look like under the microscope. So in the middle here, these are actually real organisms. This is called a diatom, a ciliate. And you'll find them in pond water if you look at pond water under the microscope. So life under the microscope looks absolutely incredible and amazing. And here's just a couple examples of it. So I look at cells under the microscope, so it's kind of cool to like, show what it actually looks like to people so they have an understanding of what biologists are working with. So in the top left, these are called the dancing diatoms. I've actually seen these in real life under a microscope and I looked at pond water and they're really cute. And then we have my picture right there. We got the diatoms, which is a single-celled algae, and they got the walls made of silica. And then at the right, we have a protist. And you can just kind of see some of those intercellular interactions happening in these pictures and videos. So I'm just trying to lay out, what biologists are looking at and how complex just the most simple cell is, and the diversity of life even under a microscope. All right, so this is one last slide I think I have on it.
Well,
We have over here. What'd you say, Rix?
I don't know. I don't know if we're seeing the same things you're seeing. I'm just on the first slide, and we're looking at the program. It's not full screened or anything. So you
Okay,
might be sharing the wrong screen. I don't know.
this happened to me before. I'm not sure if it's updating. Can we start
You know
over?
what it is? You gotta click on share whole screen
Oh, okay.
on the right side.
Okay. Yeah, you don't have to end it. Just go ahead and change which screen it's showing.
I might have to end it for
Same.
one.
It's on the same menu that you just went through.
Okay, so what do I put? Share my screen. All right, I got share stream audio, stop streaming, change stream. Is that what I push?
Change, yeah.
And then I
And then
push.
you select full screen. That's Discord.
Okay, give me 1 second.
Yeah, click that green button again. Change stream. And entire screen, top, middle.
Perfect. Are we
Cool.
good?
Yeah, go
Yeah.
start real quick,
Okay.
please.
Yeah, Riggs, can we start the stream
Yeah,
over?
it's perfect. Thank you.
Okay. Thank you. All right, the science. Okay, so I've already went through all this basically. This is just an intro to biology. Study of life and living organisms. Then we got the hierarchy right here. All living cells are made of these things. Single-celled organisms. Here's some videos. We got the dancing diatoms at the top left. And then we have the diatoms in the middle arranged to make art. And then we have a protist here at the right. So I'm just showing you guys how amazing life looks under the microscope. And I took this picture in the bottom left. So, if you get a microscope and look at pond water, you'll see something similar. But these are real things. Biology is not all completely, fake. So I just want to show
Oh,
some.
wow.
Yeah. So, oops. All right. So here, yeah. Let me move on to the next one. Right here in the middle, they'll actually make proteins fluorescent in order to track what's happening in the cell. So right here, this is a human cell, and they got the microtubules in green, which is what I study, and then they have the chromatin in the center, which is what the DNA is wrapped up in. And so this is cells splitting to reproduce. It's how we grow and develop. So we're watching real time, these processes actually happen. Then we have a neuron making connections. This is on the left, human neurons connecting one another. And then on the right, we have a white blood cell chasing a bacteria to go and, kill it to keep us healthy. All right, so I just want to show some human cells under the microscope as well. And this is
Hey,
some
shot
that
in your microorganism. Like,
would you here's some videos I've captured of the microtubules growing so I can track it real time. And here's an animation of what's occurring and then what I see on the microscope. So I'll put them in different conditions, like in the presence of aluminum, for example, just to see how the microtubule growth is affected. So yeah, a fun fact is that microtubules are attributed to fiber optic cables in neurons. And I have some sources down here if you want to go check those out. But yeah, this was recorded by me, so take pride in that. And then in the center, this is an animation by BioVisions at Harvard, just showing what the microtubules actually look like in the cell coming together in its original subunit. So it is a very amazing process to watch happen. And that'll also happen when neurons are growing and connecting as well. All right, so this is a schematic of what it looks like inside cells. This is the internal structure of eukaryote cells. So you got the nucleus that houses the DNA. You got the endoplasmic reticulum. This will actually be used to make proteins. The ribosomes on here, which we're going to go over a little bit later. And then you have the mitochondria here, which they say, well, it makes the ATP. And they say basically this happened because it has its own DNA in it. And one cell basically engulfed another cell back in the day. And that's how we got the mitochondria. But it has its own DNA as well. And that's a fun fact. And we can actually trace back, how, some people trace back how long humans have been around due to the differences in the mitochondrial DNA and can trace it back to under 10,000 years or whatever. So that's when, humans originated and stuff. But that's another topic that I might go over later in the future. And then we got some other organelles. They each do their own functions, and it's like a little city all working together. It's absolutely amazing. This was a drawing done by Dr. McGill in the top right. This is a quote from him with the drawing. So although people are struck by the complexity and density of this environment, what we depict is actually a very dilute in its concentration relative to a real cell. So this picture in the top right is a depiction of what it kind of looks like of individual things going on in the cell. However, he says this is diluted. So you can imagine how, complex and amazing the cell is. All right, and then here's some pictures of the organelles in the bottom left, and then in the bottom right, here's Clandomonas, which is what I work with. All right, so here's some basic biology I'm just going to go over because I'm going to mention a few of these different things. So I just want to give you a little rundown, okay? So here's the main different types of macromolecules in the cell, are the types of macromolecules. So we got amino acids, which make up proteins. And then we have fatty acids, which make up lipids, which is like the cell membrane. Sugars, which make up carbohydrates, so we use for energy. And then nucleotides and nucleobases, which make up DNA and RNA. So all of these are different types of macromolecules. And something I mentioned here is the chirality of them. So this is a huge issue for original life, is like, okay, because they have to have their own types of chirality, but in nature, whenever we form these, they'll form both the left and right-handed formats. So amino acids are all left-handed. Okay, they have to all be in one orientation in order to form a protein. Same thing for DNA. They are chiral as well. They are right-handed though. So DNA is all right-handed. They have to all be right-handed to form DNA. All the nucleotides have to be right-handed. And then carbohydrates can have each type of chirality, both left and right-handed, depending on the type of carbohydrate. And lipids are mostly right-handed as well. But what they focus on in original life research is the amino acids and proteins, the amino acids which make up proteins, and the nucleotides which make up RNA. Because you have to have these basic proteins and DNA in order to have, the most basic life form that we can conceive of that we evolved from. Okay. So those two are going to be focused on a lot. And, the DNA, as I mentioned, is all right-handed. Amino acids, which make up proteins, are all left-handed. Okay, so this is a definition I put on the right, chirality. They means that they cannot be superimposed on each other. So if you take your hands, they are chiral. You cannot superimpose them without like flipping one, right? So that's an example of, chirality, and they use the left and right-handed examples to depict this. All right, to be homochiral means, like the proteins are all left-handed, for example. So that gives them structural integrity and preserves their function. So if you get something heterochiral, like in nature, whenever you make these amino acids, you're going to get both left and right-handed amino acids in nature, kind of like they got in the Miller-Urey experiment, which is the most cited evidence for abiogenesis. But that can't be used for life. So even their best evidence is still not usable for life. So we're going to go over that in a minute. All right, so here's the molecular composition of cells, just so you can get an idea of the size of it. How many of these molecules are making up, a cell, right? So the smallest cell that we have, they say, is 100 billion atoms. One yeast cell has 42 million protein molecules. The shortest DNA strand that we've seen in nature is from Mycoplasma genitalium, and they have 580,076 base pairs. So that is a very long DNA strand for being the shortest type of DNA. And I think this bacteria is found in the reproductive organs. So it has the shortest DNA strand, still having 580,000 base pairs. All right, 580,000 of these nucleobases lined up. All right, and they form the pairs. That's why it's like the double helix down here that you see. So anyway, 580,000 of those. And then you have 507 genes, which one gene, depending on how it's spliced, can code for multiple different proteins. So I just tried to give the most basic life forms just to show you how complex and amazing even the most simple cell is. All right. So here's a fun fact. They actually made this GMO bacteria. They tried to take as much DNA out as they could to see how many genes and proteins they would need to still survive. So they took this bacteria, the mycoplasma, they spliced it out to see how much DNA would still be required. They needed 438 protein coding genes. All right. And we see up here normally it has 507. So they're only able to get rid of a few of them just to still stay alive. And then 531,000 base pairs. This is like the minimum that we could get for it to still function. All right. And then just some fun facts about the human body. It's 65% oxygen, almost 20% carbon, around 10% hydrogen, and 3% nitrogen. Okay. Now here's some big questions, right? How does it all work together? How do all these macromolecules work together? Where does it store its information? And how does it use the information it has stored? So this is what we're going to be getting into, okay? All right, so
Okay.
this is called the central dogma of biology, okay? So the word dogma was actually a misnomer, as it implies an unchangeable belief, but they still call it that, right? But yeah, having dogmatism contradicts the scientific method, so we don't like using the word dogmatism in science, or you shouldn't be using it. You shouldn't have dogmatism. You should always be open to changing your mind. But yeah, we'll see that even with the evidence working against them, they're still, they're never going to take up a creationist perspective in the mainstream. That's fine. But I'm just going to lay out the evidence for you guys. All right, so this is the central dogma of biology. Basically, you have DNA, which houses the information, which gets transcribed into RNA, and then translated into a protein. Okay, and proteins are what goes and does all the work in the cell. So we have the information stored in the DNA. We have the RNA, which leaves the nucleus in the cell. All right, then it gets translated to a protein. You need like, I think it was around 50 proteins just for this one process to happen, a translation. So for even one protein to get formed, you need like 50 of them. But anyway, this is just the central dogma of biology, DNA to RNA to proteins. All right, so genetic information flows from nucleic acids in the form of DNA and RNA to proteins during gene expression. All right, this is very basic to biology. And here's a picture of DNA I was able to find, because some people, kind of skeptical of DNA, especially, people in these conspiracy groups can kind of be skeptical. Is DNA real? What is it? I think we can deduce that there is some type of genetic material just because we can see traits that we inherit from our parents, and that has to be carried somewhere. We have to house this information somewhere. We know that we start off as one cell. Our parents come together, and you had a sperm and an egg for humans come together and, share genetic information. We start off as one cell and we grow and grow and grow. So somehow that genetic information has to be carried on from your parent to offspring, and that happens in the form of DNA. Okay. So that's a central dogma, DNA to proteins. All right, so the origin of this whole thing, right? Was it just a random cosmic coincidence? All right, so the scientific community hypothesizes that life originated through a random undirected process known as abiogenesis. So abiogenesis is the idea that life arose from non-life. Okay? And so here's a little fun fact I have on the right. Panspermia is the concept that life may have originated elsewhere in the universe and reached Earth through space travel. So Watson and Crick, the people that actually discovered the DNA helix, even though it was actually a woman and they kind of stole her research, but they were the ones that published the DNA double helix and discovered, what it is that's holding the genetic info. After they saw the DNA and how amazing it was, they said, this must have a designer. But since, this is just a story how it goes. Since they were atheists, they didn't want to attribute it to God. So they said, oh, this must have came from some other planet. And that's where the concept of panspermia came in. But there's a lot of really big issues with panspermia. And by the way, it doesn't answer the question of how life originated. It just pushes back the problem. So panspermia doesn't actually solve anything for origin of life. But that's just a fun fact. They knew that this was intelligently designed, but they just wanted to attribute it to space aliens or something, even though those aren't real. Anyway. All right. So here's the history of abiogenesis. So in 1859, Charles Darwin proposed the theory of evolution in his book called On the Origin of Species. All right. That was in 1859. 1870, the term abiogenesis was coined by Thomas Huxley to describe the process by which life could arise from non-living matter. 1871, Darwin, they were apparently putting pressure on him because they were so happy with the theory of evolution that they wanted a theory for the origin of life. They were just kind of getting ahead of themselves. And so Darwin, apparently, from what I've heard, was pressured to say, that life could form on its own as well in some warm little pond, quote unquote. So he said that in 1871. And then in 1929, this is when the idea of the, primordial soup came about, where it was just a mixture of organic compounds in early oceans energized by lightning or UV light that could come together and somehow form life. All right. So on the right, we have abiotic synthesis of reactions on early Earth. So they have, and they still say today that organic molecules come from space. They go to the prebiotic soup, the chemical soup, and then make these polymers to make proteins and DNA. And that led to the origin of life, which leads to RNA world, which is like the most common theory that is used now, and then proteins and DNA and, the modern cell. Okay. Whenever Charles Darwin actually came up with this theory of the origin of life, they barely knew anything about the cell. And I was able to find a quote from around that time in 1868 It was called Mr. Darwin's Hypothesis. So this is a quote from a book that I pulled around the same time. Fortnightly Review. This is actually a magazine that was published, and this is what it says about the cell. The simplest form of organic life is not, as commonly stated, a cell, but a microscopic lump of jelly-like substance, or protoplasm. The definition of a cell is, quote, a nucleus surrounding protoplasm. So they were literally just thinking the cell's like some nucleus and it's like, protoplasm. That's all they thought there was to it. Like they didn't fully understand the complexity of life at this time, but I just want to give you like an idea of what they thought about life at the time when they came up with the theory that life could form on its own. They thought it was just, this like ball of jelly-like substance. So they didn't really know about the complexities within the cell. All right, so it was 70 years after abiogenesis was first proposed that we could see the intricacies of the cell itself. So up here is actually a flagellum sliced in half in the top left picture. And these are microtubules orientated in the, 9+3 format to get the flagellum. And then we have, some other organelles I put pictures of, but only much later, whenever we got much better microscopes, could we see the intricacies happening within the cell. And it wasn't just a ball of jelly as they thought it was. And so, yeah, if you think it's a ball of jelly, of course you can think chemicals can come about just to form life. But now we know that that doesn't happen. All right, so the electron microscope was big in, looking at organelles in particular, but now we can actually see these organelles in living cells like I do, like I showed on the microtubule video. So you don't have to like fix the cells or kill them in order to see them now. And since we have fluorescent microscopes now, and we can make different proteins fluorescent, we can actually look at their dynamics and interactions without having to fix the cells like they do for electron microscope. But the first electron micrographs were put out in 1945. This was the first time that we revealed subcellular structures, which would explain why life can do what it does now. But we don't fully, understand how it all works together. We can't like model it out, but we can still understand that it's a lot more going on than we can actually see. And, I track the microtubules in particular and their dynamics. But anyway. All right, so only much later after the theory of abiogenesis did we see that there's a lot more going on here. All right, and I went to learn about the current state of knowledge on abiogenesis. So I went to my university, was looking through some of their textbooks that they give out to freshmen, and this is basically all that they mentioned on it. They don't really go into much detail on it in the biology textbooks because, like I said, the evidence is pretty shaky and they can't even come to a concise theory on how it formed, how life formed. But all it mentioned was the Miller-Urey experiment, and I'm going to go over that in a minute. And what actually happened in the Miller-Urey, this is like the most cited thing for evidence of life forming on its own. So I'm going to break that down. This is basically all that it mentioned in the textbook that they were giving out to freshmen. This is the most popular textbook, the Campbell's, that they use at universities. All right. So here's like I said, the most cited experiment for abiogenesis is this Miller-Urey experiment. So what they did, so they were attempting to simulate Earth's early conditions. They mixed gases, methane, ammonia, and hydrogen in a sealed chamber. They added water and heat to create vapor. So you can see that over here. This is what they start with. Can y'all see I don't know if y'all can see my cursor or not, but I think y'all can. All right, so they got this water down here.
Yeah, I can
Cool.
see it.
Okay, so down here they have the water, methane and carbon dioxide, I believe. Let's see. Oh yeah, so methane, ammonia, and hydrogen. Okay, they put that in here. And then they basically got it to evaporate, and they had this water vapor going up this tube. Then they struck it with lightning. All right. And then they did condensation or precipitation. They cooled it off so it would form water again from the vapor. And then they collected it over here. All right, and this is what it looked like afterwards. People have actually replicated this experiment. So it looks like after they do this whole experiment, they get this like tar-like substance on the bottom of a flask here. And then here's the apparatus where they basically struck it with lightning. So just in this breakdown right here, we got to start off with a very set of particular conditions and starting components of the methane. Let's see. Let me go over it again so I don't mess it up. Methane, ammonia, and hydrogen. They heated it up, struck it with lightning, cooled it off, and then they collected it. All right. So this is a setup of the Miller-Urey experiment. And here was their results. So here they used paper chromatography to basically separate their results. They found a few different amino acids, all of which are extremely tiny. We got the glycine, made of like 10 atoms, the beta alanine 13 atoms, and alanine 13 atoms. This is their results that they got after separating the components that were present in the flask after they did the experiment. All right, so amino acids only composed of 2% of the residue after doing this experiment. And this was all they published, by the way. I went and found their original paper. And by the way, not to mention, remember earlier we went over chirality? So the amino acids have to be all left-handed, but in this composition, they were actually left and right-handed. They were heterochiral, so they could not be used in life. So even their best evidence still cannot be used because it's both left and right-handed amino acids. So yeah, that's just going over what they found, and they found they found 3 of them, but later I think they isolated more amino acids, but they're not useful to life because they're heterochiral. All right, so they have published some criticisms of this experiment. Number 2 is kind of disputed, but the rest of them pretty much still stand. Like how everything was every concentration of methane, ammonia, they were all very carefully selected. People say their atmosphere wasn't completely accurate, and that lightning would have like destroyed organics, like if this was to continue, and organics that were present, if they were to go through this environment, they would be destroyed due to the lightning. And then also, let's see. Oh yeah, other molecules that were formed in this tar, because there's only 2% amino acids, would be destructive to life. Afterwards, they carefully selected and separated the amino acids from the mix, but in nature, you wouldn't have that happening. It would just be what it is. You just have to work with what you got. So they carefully selected amino acids, and they're still both heterochiral, so they still wouldn't be useful for life, which is what the next point mentions. And then formaldehyde and cyanide were present in the tar that they made, which are destructive to life. All right, so some people say that they weren't accurately even representing the atmosphere present at the time, that it had much more oxygen than they claimed present. I don't think they mentioned any, because if there was oxygen, everything would just be oxidized. So, and then also, they posit there was very low concentrations of NH3, and the similar experiments in oxidizing environments yielded no results. So if they were to have this oxygen present, it would have just oxidized everything. It would be done for. All right, so in the Miller-Urey book, Their Planets, Their Origin and Development, it stated, quote, "The primitive oceans would have been approximately 10% solution of organic compounds. This would provide a very favorable situation for the origin of life." But actually, even with all the life present in the earth, this was a university in California that actually measured the amount of organic matter in the oceans and found only 1 to 3% organic matter. But they were assuming that the ocean was 10% organic matter. So in their highly selective environment that was trying to mimic early Earth, they were obviously doing what would best make their results, and it wasn't even reflective. Like what they thought the oceans were not even reflective of what they are now. So before life formed, we could see it was probably even less, maybe under 1% organic matter rather than 10%, which is a big difference. All right, so here's a quote. A genetics researcher, Dr. Albert Brancombe, and he works for the Institute of Genomic Biology at the University of Illinois. So he said, quote, "We claim that living systems evidence a preoccupation with maintaining chemical specificity, not having just the right molecule to hand, and especially not having all the wrong ones. So we need them not only to have the right things present, but not all the wrong things." Remember how I mentioned it was only 2% amino acids? So you have a lot of those wrong things present. Yet in any solution chemistry or any chemistry not managed by a macromolecular machine, acting specifically to affect a particular transformation needed, the latter wrong ones invariably overwhelm the former right ones. So without our machinery in our cell, you're going to get a lot of these wrong and bad molecules come out of these random sporadic reactions where they just zap chemicals with lightning or electricity. And that's what we saw happen in the Miller-Urey experiment. And so this leads to the tar problem. All right. This is actually an origin of life researcher, a very prominent one too, who's made significant advancements, quote unquote, in the origin of life field. So he said, the building blocks assumption is simply dead wrong. As a simple thought experiment should suggest, if you make up any mix of building block products you like, however rich, energize it in any way you like, wait as long as you like, nothing we assert having any claim of any sort of being lifelike will ever arise in your flask. Not ever. Instead, the chemistry therein will more or less slowly progress towards Benner's tar. All right, so he's saying, he's kind of coming against this experiment in a way by saying, you're not going to see any life come about by zapping molecules. It's going to be a very slow process because whenever you do stuff like this, you just get Benner's tar, and the tar we saw at the bottom of the flask. All right. So the next thing I set out to do after, looking into their best evidence was look at their more common theories on the origin of life. So what is their concise model? Surely by now, with 100 years of research, they would have some sort of model. So in this paper, this is a recent published paper. They used a computer code to infer the order of the addition of amino acids to the first cell. And some of the things that were quoted in it were really funny. So I basically looked at recent papers published, and this is so funny. Like, some of the things they mentioned possibly derived from extraterrestrial sources, such as asteroids or comets. They're still having to invoke outer space and what's going to come from it in order to have some of these basic compounds. They talk about the hydrothermal vents, amino acid from asteroids. Like they're still trying to, talk about asteroids contributing these things. But even on the asteroids, they're still heterochiral, so they're not actually usable. They talk about the subsurface of the ocean and hydrothermal vents. And let's see. Here's a funny quote. Perhaps the biggest mystery is how sequences such as the common ancestor of LIV tRNA synthetase, which were translated via alternative or complete genetic codes, ended up being recoded for translation by the direct ancestor of the canonical genetic code. So we're going to get into these problems with the tRNA a little bit later, but did you want to add anything, Dan? No? Okay. Whew, I got to take a breath. A lot of talking. Yeah, that'd be great. Okay. So I was looking for the most concise scenario for their origin of life theory. And so this is what I came up with, right? Because it's so funny. For every different molecule, they have to have a whole different set of circumstances and environmental conditions. So here was the summary of the current abiogenesis scenario. So between the edges of a hydrothermal vent, ocean water, and volcano rocky pores formed nature's own chemical laboratory. Each pore offered a unique microenvironment, stockpiling organic molecules. Lipid components delivered by meteorites crashing into the earth provided raw materials for primitive membranes. In another location, lightning strikes while UV lights radiated shallow pools, driving critical reactions, especially for RNA. So right here, we already got, lightning. We need UV. We need this corner of a volcano rock and micropores, which each have their own environment. So, like, this is funny because whatever the cell doesn't do, whatever we don't have, we have to put onto the environment. So everything that we've went over so far is critical to life. But if you don't have the machines within the cell doing it, you have to push everything to the environment to pick up all that weight. So then at the end of the day, what they have is an unreasonable amount of pressure on the environment to do all these different things that the cell is not there to do. So we have the clay surfaces that are needed and acting like molecular assembly lines, aligning the right concentration of molecules in the pore of a rock, while magnetic minerals and crystals align them to get them in the right configurations. This is their, quote, solution to the heterochirality problem. They want to invoke magnetic minerals and crystals. Then they want to invoke clay for bringing all these, monomers together to form polymers. So they're having to invoke all these crazy things, and they can't get one concise theory because every problem they're coming up with a new situation to fix it. Then you get this crazy storyline. So whenever people bring up the theory of life and how it formed, I'm like, okay, give me a rundown, because you have to invoke so many different types of components and environments for every little part of the cell, in every molecule. Then you have wet and dry cycles that are needed in the pools. Then you have RNA migrating from shallow pools to the hydrothermal vent, tiny lipid bubbles emerging and encapsulating the RNA while still being able to hold the contents and transfer materials from inside and outside the cell wall without breaking down. So it began migrating between these environments, scavenging components and energy wherever it could. Through this interplay of hydrothermal vents, meteorites, lightning, UV light, and drying pools, wet and dry cycles, the beginning of life was first born. So this is such a comical storyline, like of how much they need already. And this was the best rundown I could get based on their recent, theories. So here's an example of a few different things that they have to invoke and what problem it's actually solving. So you need this more, I can't even pronounce that word, this type of clay in order to form RNA on it, for RNA polymerization. And this will happen in the tidal pool or volcanic ash area. Then you need this pyrite, for pyrophosphate synthesis near hydrothermal vents. You need iron sulfur minerals to make amino acids near a hydrothermal vent. And then you need borate minerals, for sugar preservation, in some sort of evaporative environment. So this is just a few different things. The RNA, the pyrophosphates, the amino acids, and sugars, and their solution to them. So can you tell me where anywhere on Earth is going to have all these different like components and environmental conditions? Like this is so crazy having to put all these different things together. Like you're just putting way too much pressure on the environment. Um, but up to now, did anybody have any questions? While I take a sip of water?
Well, it just seems like um, all these things need to be present and available and bioavailable at the same time in order for any of the processes to be successful.
Yeah, I
Is that
mean, we're
right?
just talking basic components. We're just talking about how we get these basic components for life, like
But
amino
not
acids.
just that, but the instructions to
Yeah,
know what to do with all this
we're
stuff.
gonna get to that later. Yeah, I have that
Okay,
later. I'm
I got
just
a
talking
question.
about. Yeah, go ahead.
Why, why, why are things that are ambidextrous useless? I don't understand that
Ambidextrous.
part. Well, you're like they're
Yeah.
both
So
right and
The
left-handed, so
chirality.
they're useless. I don't. Can you explain why they're useless?
yeah, I can. I have a slide on that back here.
I'm sorry. I don't know if
No,
that's a second.
that's a good question. So they all have to
Mhm.
be left-handed. Let's see.
Is it
Because
like a heterochiral or something?
yeah, that is heterochiral, left and right-handed, but they have to be homochiral. It provides
Mm.
structural integrity and preserves function. And not to mention, whenever you have these polymers come together, you're basically adding one on the other. So they come and attach in a row. But imagine like stacking up chairs, but like one of them's facing the wrong way. The chair is backwards. Do you really think you're gonna get a nice pile of chairs if one of them's backwards? Like it's gonna mess up the structural integrity.
Perfect example for the guy in the wheelchair. That's
There
a
you
great.
go.
I completely understand that.
There we go. Okay, I'm glad
What a great
you.
teacher.
Why does, why does that point to God though? One chair is like not
That's
in
Seriously, that was a
that's
good
another
example.
question.
Yeah, well, what I'm laying down here is just that, we can't reproduce not nature actually doing what is required for life. And I'm gonna get to a couple paradoxes in a minute. So let me just keep moving forward.
Hold on.
And
I got one more question. You opened it up for
I will. Do you have like a specific question about what I
yeah,
want
it's a
to?
specific yes. What you're pointing out is nature, but you're not really showing us God doing this. So like can you show like Adam, you know, when God made Adam from the dirt or dust? Are you ever going to show that or no? Because that's a positive claim.
God did everything, you know. So I think that God could definitely make life. I think that at the very least we need intervention.
So you're basically antagonizing or criticizing the current abiogenesis or science model the same way that flat Earth criticizes globe Earth.
Okay.
You're doing the same thing. Yeah, I know. So
What
false
analogy is this?
is independent of replacement, right? That famous. You've heard it before.
Well, I'm gonna get to the positive evidence for an intelligent designer, but that's the last
Cool.
part of the presentation. Right now, I'm just breaking down their theory as it stands.
Right. That's what I'm hearing, but I haven't been here the whole time.
It's all good. Yeah, so right here I'm going to be going over a few compatibility issues. So for the RNA and the proteins and DNA are, you know, even if you just look at RNA and proteins, these are going to need different environments and different conditions. Like the protein synthesis needs greater than 100 degrees Celsius and a dry environment because you don't want hydrolysis to occur for protein synthesis. But you know, RNA, you can't have RNA getting that hot; it's going to break down. You got 55 to 85 um, degrees Celsius, especially in wet and dry cycles. So yeah, so like these like environmental conditions that you need for each are incompatible. So at the very
Right.
least, you can see the most
Can I
basic one is formed in
ask
different
something?
locations.
Can I ask something, Taylor?
Mm-hmm.
Okay. So are you saying that the environmental conditions based on the current presented representation of the beginning of the earthular cycle in the universe, the cosmos, is incompatible because the heat at the beginning is so great that these formations are impossibly placed in proximity to each other? Meaning we're talking about a 10 to like the 120 like you're not saying you're saying it's so impossible because of how outlandish the fine tuning it is. So there's like God at the period of it no matter what.
Oh, no. Right here, what I'm showing is that like you need for protein synthesis it to get to a certain temperature that would not be compatible with RNA, yet you would need both of these to be in the same location for life to form. That's what this chart is going over. It's the incompatibility paradox.
Meaning that there was a duality of environments going on at the time
Yeah,
that can't
yeah.
exist. Okay. So that's like Schrodinger's cat, dead and alive at the same time. Paradox. Okay.
Labeled it. When you need each other but cannot coexist. So you got the RNA, its temperatures, and their optimal pHs are different as well for protein and RNA synthesis. You need to have let's see, hydrolysis will occur in the RNA, yet you need to have these like wet and dry cycles happening. But hydrolysis will occur with proteins as well, whenever their amino acids go to form into proteins. Like you need an aqueous condition, yet you have like hydrolysis happening. And like especially whenever you go to have RNA and proteins together, their conditions that they need to form are incompatible. Right here, I mentioned one like with the temperatures, like RNA can't get that hot. However, in order to take the pressure off the, cell, because there is no like enzymes causing these reactions, you have to have it get to like a crazy high temperature to have the energy for these, protein bonds to form. But that would break apart the RNA. So like, but you would need both of these to get to the central dogma of biology from DNA to RNA to proteins. Like you'd have to have these coexist, but they're incompatible, like their conditions to form and stuff. You know what I mean? So that's kind of what I'm showing here. And then protein formation happened in like volcanic surfaces because they have to have like super like basic conditions. But RNA synthesis would happen in their latest theories in like some tidal pool exposed to UV that, in the paper they said it could have got its energy from like meteorite strikes. So yeah, I mean just they acknowledge that this would happen in different locations or at least different environments, which is why they have like the micropores theory in the volcanic rock. So like, yeah, this is just the incompatibility paradox when you need each other but cannot coexist. Let's see. Okay. Okay. Yeah, and then the RNA needed UV light, but that would rule out the deep sea hydrothermal vent, which is the current leading theory for protein generation and formation. And so here's showing like their stability over time, RNA and protein stability over time. This is in a matter of days, right? So remember, they would need wet and dry cycles. So here's wet and dry. So in an aqueous condition, you got RNA lasting less than 1 day and like, around room temperature. And you got proteins lasting around 20 days. In aqueous hotter conditions, like maybe like a hydrothermal vent, you got both of them lasting maybe 1 day. You have the highest compatibility in dry environments and room temperature. But then again, you need the wet as well because the wet and dry cycle is required for the protein formation. And then at dry, high temperatures, you can see the proteins are a little bit more stable, but not the RNA. All right, so here's another compatibility problem. It's called the Maillard reaction. So amino acids will react with the sugars, like a ribose, which is a component of RNA, in the Maillard reaction. So they will cross-react with one another. And we're just going over the compatibility problems with just basic proteins and RNA because that's what they say had to come first for life. But if they're left in the environment with one another, they can actually cross-react. And one of the reactions is the Maillard reaction. Amino acids reacting with the sugars on the RNA. And this would deplete the available ribose for RNA synthesis. However, they must have coexisted, in the abiogenesis scenario because you have to have that RNA to protein translation happening for even the most basic life forms. So somehow they were able to come together and do all this stuff without cross-reacting. All right, let's see. And here's another example. So lysine, arginine, and histidine significantly suppress RNA polymerization by competing with the nucleotide binding sites. This competition can lead to mixed or undesired polymers, reducing the formation of RNA. So you got these amino acids hopping up in there. They're supposed to be just used for proteins, but without any compartmentalization like we have in the cell, these are just going to be cross-reacting all over the place. So I don't know how that ever was worked out, but anyway, they must have coexisted, however they cross-react. Here's a translation paradox of magnesium. So magnesium concentrations over 2 millimolars will suppress nucleotide incorporation into DNA. So you can't have magnesium that's too high because you can't get these nucleotides to form. However, you need higher concentrations for amino acid chelation of about 15 millimolars total concentration. So 15 millimolars total concentration will increase amino RNA folding and stability. And they'll have around 2 millimolar free magnesium. But wait, we just read that anything equal to or over 2 millimolar will suppress nucleotide incorporation. So here's another compatibility problem with free magnesium. If you have what's needed for RNA folding, then that's also going to suppress other things like nucleotide incorporation DNA. So, everything in the cell, as I mentioned earlier, is sectioned off. So you won't have these cross reactions, but in an open environment, they're just going to be cross reacting all over the place. And then the phosphate problem. This is so funny. This is one of my favorite slides because it literally calls NASA out so hard. So there was a problem with organic phosphate being present in the early Earth, and so phosphorus is used for the DNA and RNA backbone. So you have to have that bioavailable phosphorus. However, a lot of phosphorus without organic life present would be locked up in the rocks, and it would already be bound to other things. So how did phosphorus become bioavailable, and how did the cell get over this issue? Like, how did life form while the phosphorus needed for DNA is locked up in, chelated in the rocks? So NASA actually came out with this paper that would kind of solve this issue. I don't know if they addressed the issue particularly, but they were using this as an explanation to solve the problem for original life. So they were saying that they found this new form of life that it was using arsenic instead of phosphorus as the DNA backbone. So that was a huge deal. Like, there's another type of life form that we found. That's crazy. So they were like doing all this hoopla about it. It was pretty crazy. But when people actually looked into the paper, they were finding like that there actually was phosphorus there. The arsenic was like contamination or something. And they posted a lot of papers saying scientists say NASA's new arsenic form of life was untrue. And then NASA have since like came out and redacted this paper and stuff or whatever. I don't know, but they fired the person apparently that was working on the research at their, funded biological research site. And yeah, they've been basically called out by the scientific community. I guess the biologists don't play around. I don't know what's going on with the physicists, but they don't play around over here. And yeah, their thing was untrue. So this leaves another problem with the phosphate problem. If all the phosphate was bioinorganic, how did we have it for the DNA backbone? So NASA falsely claimed that life forms could get around the need for phosphate by using arsenic as a DNA backbone. But now, from what I've read, they're just appealing to comets and asteroids to bring in this, this organic phosphate or whatever. But yeah, this is a funny thing if you want to look into NASA being called out by other scientists. All right, here's a list of a couple of paradoxes that I put together on the origin of life. Now, as I mentioned earlier, they'll do some crazy backbends to try to get over these problems. The problems still exist, but they're going to have some other crazy environmental scenario to try to get around it. Like, oh, it happened on clay, or, it happened with pyrophosphate and pyrite and crystals or whatever. So these are still problems, but just know that they try to like make solutions, but the problems still stand, and them trying to make solutions is how we get some crazy environmental condition that life they say had to form in. But anyway, so this is the water paradox. Water is commonly viewed as essential for life. It absolutely is, for many different reasons, which I don't have listed here. So are biopolymers like DNA, RNA, and proteins. However, these biopolymers are corroded by water. For example, hydrolytic deamination of DNA and RNA nucleobases is rapid and irreversible, as is the base catalyzed cleavage of RNA in water. So we find that these polymers will break down in water, like RNA and DNA. They will break down, especially water that has any contaminants in it. However, the cell needs water. Water is like the universal solvent, and it allows multiple chemicals to come and interact with one another. If everything was dry, it wouldn't be doing anything. Basically, everything's reacting because it's in water in a condition, a solution that, allows them to come and cross react with one another, which, in life, you'll have a lot of happening in the cell, as long as it's not in a destructive way, like we see without the compartmentalization of the cell present. But here's the paradox. Yeah, the water paradox. You need water. However, with water present, you'll get things rapidly and irreversibly breaking down. So RNA requires water to function, but RNA cannot emerge in water and does not persist in water without repair, especially contaminated water like we would see in the, open atmosphere. But that's the water paradox. We also have the energy paradox. This is a really, really important one, and it's really interesting. So energy is needed for life. It's what fuels the reactions. We have ATP. ATP is the currency for energy in the cell. However, raw energy without proper ways to process it will cause destruction. So, for example, have you ever tried to charge your phone on a charcoal grill? I mean, I'm sure you can imagine what would happen, right? Your phone is just going to break. There's energy, but it has no way to process and use that energy and to convert it into a usable form. So energy is needed for life, yet without the mechanisms to actually convert it to usable forms like ATP, which is actually an incredible process, it's just going to be destructive. So it's like putting a Tesla charger in a gasoline intake hole. It doesn't have the machinery present to process energy. And the early life forms, and how are we going to form these molecules needed for life? They're not going to have any ways to process the energy. So any energy, while it is still needed for certain reactions, is destructive. So it's very counterintuitive and paradoxical. All right, so here's the UV paradox. So in the papers I read for like where RNA is going to get its energy to bond, they use UV light. Okay, so UV is crucial for the formation of key organic components necessary for life. However, if unchecked, that UV radiation is going to break down and destroy the very molecules it helps to create. So, for example, in the presence of UV light, DNA will form pyrimidine dimers, and also UV light will photodegrade organic phosphorus compounds. So, they're invoking UV light, and it's needed for some cases. However, unchecked in the environment without mechanisms of repair, you're just going to get destruction. Just like the energy paradox, you just get destruction without these mechanisms to repair and keep it in a homeostasis type manner. So there's no homeostasis without life. All right, here's the hydrothermal vent paradox. So they still use the hydrothermal vent, as I read earlier on some of the latest papers on abiogenesis. They're still used and proposed for protein synthesis. However, they need the high pH and a very basic condition for proteins to come and form, the amino acids to bond to form proteins. However, the alkaline vents strangle out carbon life, and it is destructive to RNA. So a study published by Nature Geoscience revealed that hydrothermal vents conditions dissolve free organic carbon compounds to carbon dioxide. They wrote that even the most resilient organic molecules eventually broke down as they circulated through the vent systems. So it's funny, like, yeah, you're strangling out all the usable carbon. However, we need these hydrothermal vents to form proteins. So it's just another contradiction. And the heat paradox. So you got heat as a catalyst. Heat will accelerate chemical reactions, providing energy for forming complex organic molecules. However, heat is also very destructive. It will break down delicate biological molecules like RNA and proteins. So here's your heat paradox. You need the heat, but it is destructive at the same time. So everything has like a checks and balances, especially within the cell. I mean, we don't really need heat to form our bonds, our chemical bonds now, because we have enzymes. But back in the day, when there was no life, as they say, they wouldn't have enzymes to come do all their nice work for them. So they have to put all the pressure on the environment. That's why you get to unreasonable needs for unreasonable heats. And then that heat will ultimately be destructive as well. So it's paradoxical. Did you want to say
Anybody
something?
anybody who's cooked an egg, I mean, once you heat protein up, it doesn't go back. So these are like very delicate materials, and that's just one thing. There's like a million of these things. It's crazy.
Yeah, but remember, they don't have enzymes to come make. So these reactions and bonds, they need energy to bond. They don't freely bond. This is why we have enzymes to come and bind things together. Okay? But when you don't have enzymes, where are you going to get the activation energy needed for this reaction to occur? So, of course, they push it on the environment. This is why they need heat for protein synthesis, for example. They need the heat to make these bonds happen that aren't going to happen normally.
So they need the heat, but they don't need the heat everywhere. They need it in precise places.
Yeah, at certain times. Yeah, we just need heat
Yeah.
Well, at
here.
the same time, at
So-
a different time as well, just for this exact moment, it popped into existence just long enough to be there, but then it wasn't again at the same time.
Right.
Yeah.
So like these little the ends of the proteins, it's like they have a little like welder at the tip, just for the precise moment it needed to be done.
All right, we're done. Turn off the heat.
So what if you could like have a place where you could keep the heat constant or static, like say when you look at a fruit, right? If you look at a orange, right? And you take off its peel and then you even open that little like protective layer, it has little compartmentalized protective layers for the juice inside or the life inside to stay alive and not be destroyed by the sun.
There is
Because we
no
all
life.
know
the formation of life. So we can't invoke life to explain the formation of life because there was no life.
You're talking about a system with enzymes, man.
There's a system inside that orange.
Of
That-
enzymes.
Yeah, but the
Cool.
orange is, but we don't have
But
life.
there's a protective layer there. Do you discount that?
Where do we see that in the environment without life? That's what we're talking about. We're talking about the formation of life. So there is no life, there is no protective layers. This idea of the oranges having compartment, that's cool and all, but you can't.
Organisms.
You're
Yeah,
using
the
life
organisms
as an
reside. The organisms reside in like fluid membrane, like capsules, dude.
there is
Where
no-
the heat wouldn't get to it. Unless it did, then it's gonna kill it. Just like we see right now. Like if
What
you
builds
stay
your
out in the
membrane
sun,
without life? Where is your membrane without life?
Yeah, this is what. Okay, let me go back
You're
to
invoking an analogy of life for to have unlife. Like you're saying this analogy, which is life, is analogous to not life. It's like they're not analogous. One is death and one is life.
Okay, so this is what I'm going over right here, the origin of life. All right, so the scientific community hypothesizes that life originated through a random undirected process known as abiogenesis. Abiogenesis is the idea that life arose from non-life. So we are trying to get the first life form. That's what I'm going over. So there's not going to be any, natural selection. There's not going to be any, homeostasis or coping with the environment. There's none of that. Right now, we're just talking about chemicals forming life. You understand this definition right
to
here?
answer one of the most difficult questions in our infancy. Like, do you agree that science isn't at its apex, like set of knowledge right now? Like, we just barely started looking at these things.
I have a slide on that later. So it's actually like, remember earlier, whenever they came up with the theory, what they thought cells were? Well, I'll just tell you, they thought cells were just balls of jelly. They thought they weren't complex. They just arose on their own in chemical soups. But now, the more we learn, we see that this is more and more impossible to conceive of how, this idea of
We just
abiogenesis.
don't understand
So
it. You keep claiming we can't ever know, but
I didn't say that.
eventually.
Well,
So I
okay,
thought
so
you were
what
done
she's
with-
claiming,
I thought you were done with.
I think
she
what
was
she's
done.
really, I think what
Oh, I
she's
didn't
really claiming, Helios, is that the more we learn about these processes and how they function, the more astronomical the numbers become. It's like
Yeah.
one 20-digit number, and then every time you learn something new, it's another 20-digit number you're multiplying by. It's like it's an impossible amount of impossibilities. So it's not that it's
just
invoking
super
God.
complex.
It's just that God is proof by exhaustion. Like there's no feasible outcome.
I'm gonna get to the
Yes.
proof of intelligent designer. So
I'm just letting you know what like the thinking is, dude. That's
Yeah,
all.
let me just go.
Okay,
I'm only
but here's another way of thought. Could you entertain another way of thought?
Dude, that's what we're doing.
go ahead.
Yeah, but you didn't hear mine. You just gave me yours.
Well, we're doing a presentation here.
Yeah, just make it quick.
But it was over, and now you're open for questions. So can I
I'm
critique
sorry,
your?
did you finish?
The questions were
No.
over, Helio. Oh my gosh.
Taylor, did you finish?
No, I'm not done.
Yeah, chill, please.
Do you have like a
We're
quick
gonna.
or no?
Yeah, I think we should just keep going. You can
Okay.
wait until you're finished.
Okay.
Ridiculous.
Here is the natural selection paradox. So researchers will appeal to natural selection to get complex machinery, which deals with all these other issues, and the compartmentalization that is required for even the most basic life form. So researchers will appeal to natural selection. However, you cannot have natural selection until you have self-replication, which requires complex machinery. Okay? So remember this, they're appealing to natural selection to get the complex machinery. But you can't have natural selection until you have the complex machinery because you're not going to have reproduction. Then you're not going to have any option for selection. There's going to be nothing to select from. So you cannot appeal to natural selection to get these machines. You have to have them for natural selection because you need them to have reproduction. And natural selection's like kind of like survival of the fittest, whatever's the most fit will survive. But remember, there's no survival if there's no life. So we're trying to figure out how life came about. So there's no natural selection that you can apply in this situation. And then I think this is the last. There's a couple more. Okay, sorry. The time paradox. This one's one of the most important things because people will invoke, oh, well, with enough time, anything can happen. All right. So time is not on their side though, because time will degrade the very thing that they're trying to make. So complex organic molecules require time to accumulate and become useful through random chemical processes. However, time is the very thing that will degrade the organic materials that they're trying to get. So you're working against the clock, dude, because as my slide said earlier, you can see how many days these things are present. Look, the half-life in days of RNA in room temperature aqueous environments, less than 1 day. All right, proteins in aqueous environments and room temperature, you have 20 days. In the hotter temperatures, like in the hydrothermal vent, you have like not even 1 day for these, maybe 1 day for proteins, 0 days for RNA. In the dry environments and room temperature, you're gonna have the longest time span of 40 days for RNA and 80 days for proteins. However, you have to have the aqueous environments in their wet dry cycles, so you cannot only have dry environments. And in the dry environments and hotter temperatures, which would probably be near a hydrothermal vent potentially, you're going to get much less again. So you're really racing against the clock. So time is not on your side, and that is the time paradox. All right, now here is the DNA protein paradox. So modern cells require proteins to replicate DNA and RNA, but proteins themselves are encoded by these genetic materials. This creates a circular dependency where the components necessary for life to rely on each other to exist, they rely on each other to exist. This will complicate the understanding of how life could begin from simple organic compounds. To overcome this, RNA world hypothesis became popular, but it falls in the same trap. So DNA is the information for proteins. So proteins need DNA. That's where their information comes from. However, DNA needs proteins because proteins are what replicates the DNA. So you're not going to have one without the other, yet they are required for each other. So what came first, the chicken or the egg? It's kind of like that problem. And then RNA falls in the same thing. DNA is transcribed to RNA and translated to proteins. So you need RNA for proteins because that's where they get their code from. Proteins get their code from DNA and subsequently RNA. Okay. And RNA needs proteins. So in the ribosomes, which is what makes proteins, you have multiple different RNA and proteins present. So you need both of them to make one RNA molecule. So this is circular. Like you need all these components to already be there, essentially, for even the most basic life form. But they all rely on one another. So how could they come about, whatever. All right, the tRNA paradox. So the translation system requires a highly evolved set of RNAs, proteins, and enzymes for high fidelity. However, these proteins are formed via the translation system. Another chicken and the egg. So even the tRNA, which makes the proteins and does the translation from RNA to protein, that also needs proteins and RNA. So, you can't win for losing, bro. You have to have it all there. Like, that's just how it works. All right. So Eigen's paradox. So this is funny. We can't have long strands of DNA unless it can repair itself. All right. And it can't repair itself without long strands of DNA. So this was posted in Nature. Okay. So he posed the paradox that without error correcting enzymes, the length of a nucleic acid would be limited because in larger molecules, mutations would destroy the information content of subsequent generations. But this maximum size or error threshold was too small to encode an error correcting enzyme. Eigen's paradox still challenges theoretical biologists. So yeah, you need to have like a long DNA strand that's like accurately being passed down in order to get repair machinery. However, these long strands of DNA, we're wondering how would they come about without repair machinery? You need that repair machinery to keep the DNA consistent without having too many mutations. But in order to have that system, you need long strands of DNA. So, it's just another circular problem where you already need it to be put there in order to have what it needs. And then the membrane paradox. This is a cool one because somebody just mentioned, oh, they came and encapsulated this and that. Okay, well, here's your membrane problem. If you did have some membrane out in the open just coming encapsulating stuff, then how is it going to have things? First of all, it's either going to need to be permeable enough to have things move in and out without breaking down. And if it's not, how are things going to move in and out of it? So then basically it's trapped in a jail cell. And the way that our cell deals with this is we have something called transmembrane proteins. So we have proteins that expand through the membrane that let things in and out of the cell. But without this, you're just going to be strangling out anything that's trying to come in and out of it. You're basically going to be killing it by encapsulation. Oh, and here's another, that was one problem with the membrane, but here's another one. So the membrane formation paradox. I need to change this name to membrane formation paradox. I'm taking little notes. Okay. Let's see. Okay, so the membrane formation paradox. So this acknowledges that membranes can only be synthesized from pre-existing membranes. As an extension, the organelle formation paradox states that membrane encapsulated organelles can only be produced from pre-existing organelles. The protein phospholipid paradox demonstrates that protein composition in organelles is dictated by the organelle's phospholipid composition, but the phospholipid composition is dictated by the protein composition. So that's just another circular problem that they have. And by the way, the membranes only come from pre-existing membranes. But yeah, this is an interesting one, the protein and phospholipid composition. All right, so this one I titled Circular Synthesis Paradox. You need 6 ATPs to make 1 ATP. Remember I said ATP is the energy currency of the cell? Well, yeah, in this citric acid cycle, you need 6 of them to make 1. You need NAD to make NAD. You need tetrahydrofolate in order to make tetrahydrofolate. You need it already present in the reaction needed to make the compound itself. You need coenzyme A in order to make coenzyme A, and FAD to make FAD, which is also in here somewhere, I believe. Yeah, right here, FADH2, right here by fumarate and succinate. Anyway, so you need these things present in order to make one of them. Just like the RNA I mentioned earlier. You need RNA and proteins to make one protein or one RNA. So it's like, where did it begin, bro? You gotta overcome that. All right, so the ribozyme paradox, I should be almost done. Yeah, so this is my last paradox. This is in the RNA world hypothesis, okay? So this is like one of their leading theories. This is what I learned about in undergrad with Origin of Life is the RNA world hypothesis. So for a ribozyme to be functional, it must be stably folded into a complex structure, much like a modern day enzyme. However, in the absence of a set-aside genome, this same ribozyme would also need to serve as a template for its own replication. So templates cannot have complex stable folding if they are to be accessible to free nucleotide and RNA primers. Clearly, the same RNA cannot meet these two mutually exclusive requirements simultaneously. In the RNA world hypothesis, ribozymes necessarily must have dual roles. They must have the catalytic activity to replicate itself and to make new RNAs while simultaneously being able to act as a template for replication. So even if we were to get one of these ribozymes, which we've never formed from nucleotides, much less from inorganic matter, we've never formed one, but assuming that they have, because this is one of their main cited evidences of its self-replicating RNA, or the ribozyme, even if you get one of these, which we have not, then first of all, is it going to be acting as a template? Like in keeping and to share its DNA, or is it gonna be actually doing its function in order to make more ribozymes in RNA? So those are just two main issues with that. All right, so this is funny. I saw this paper. The RNA world hypothesis, the worst theory of the early evolution of life, except for all the others. So they're totally dogging on this. They acknowledge that they're all bad. Like, this is why they can't come to really a theory of how life forms, because all the leading theories have serious issues. But you have to go with one of them, because look, we know now that we use DNA, RNA, proteins for even the most basic life forms. At some point, these had to come about. How did that happen? That's a big question mark. So you have to have something starting out with the basic genetic material, basic proteins. You have to go back to those basic components and use one of these theories of what came first. It's needed for all life. It's mainly like the first step in getting life is having, the central dogma, the RNA proteins, but every theory is bad. Like, and they'll even like acknowledge that, like in this paper. But I think in here they're talking about how RNA is very limited and very unstable in, in the open environment. And plus, in my opinion, it doesn't even really fix the problems, because the same issues that you're having with DNA and how it needs proteins to make it, it gives the code, that still applies to RNA. So they were trying to find an in between DNA and proteins, which is RNA, but I think it actually ends up being worse off for them. So anyway, they acknowledge though that all the theories are pretty bad. Here's a researcher, John Sutherland. He is a original life researcher. Really, I mentioned him earlier. He's made progresses, I think, in RNA synthesis or something from inorganic compounds. But that's a whole nother dog to tackle. They're basically working so hard on each individual step and component for, RNA, because it's even more complex than proteins and amino acids, like with the Miller experiment. It's way more complex. You have to have so many different parts that, how did these things come about? How the nucleotides come about? How they polymerize? I mean, so he's worked on some of that, but it's, forming RNA and DNA from inorganic materials is much harder than even forming proteins from amino acids and stuff. So anyway, I think he's done some research in that field. And this was one of his charts that I saw in his paper. So he completely concedes to the lack of any progress in explaining a naturalistic origin of life. So here's this paper right here. Degree of aliveness in the time or system complexity. So inherently favored prebiotic chemistry, that would be step 1. And he says the current state of the field's right here. And then you got energy dissipation, major system innovation, optimization of major systems, of major system innovation, necessity, contingency, boundary. And this is moving from chemical necessity to chemical contingency. And then the fully alive line is way up here. So then we got evolutionary innovation. This is when evolution kicks in. And then you got the last universal common ancestor, which we all descended from. All right, so here's the steps to make Luca, the last universal common ancestor. He says down here is the current state of the field. And it's like the more research they do, the further back this keeps going. I argue we are way back here because I don't think we figured out the favored prebiotic chemistry for any of it. But yeah, it's for him to publish this and say we're here is like very shocking to a lot of people that aren't familiar with this field. But it is true, and I would argue that we're even way back here. And so look, he says right here, a long, long way to the last universal common ancestor. So in our current state of research and in the dozens of hours, millions of dollars, hundreds of hours put into researching this, we're so far away from any like sort of system. And, he says we're still at the very bottom of this chart. But I'm glad he acknowledges that. Many people just aren't aware of that. And it's not even like, oh, with more time, we're going to get further up. But it's actually moving the other direction, because the more research and experiments we do, we see how much intervention is even needed for the most simple experiments to prove the most simple things. So much intervention needed. So it's like we keep getting pushed back on this chart the more we research it. So it's pretty crazy. All right, next chart. Let's see. Okay, so 100 years later, after abiogenesis was proposed, scientists have yet to successfully create a functional protein from inorganic molecules in a prebiotic conditions. They don't have a model. They have yet to create a functional RNA molecule from inorganic molecules. And here's a fun fact in case you didn't know. No living cell has ever been created from non-living components. not even with their best technology and the best conditions we can conceive. They have never created a living cell from non-living components. It's like we can't even do it if we tried. Like screw the origin of life research and like trying to mimic its environment. Even if we put all our best foot forward, we still can't create life from non-living materials. That's how amazing life is. Like, yeah. Right here's a quote. Studies on the origin of life. We are not yet close to achieving this end. Talking about how, the laboratory would show how life can start from the inanimate, stuff like that. They're so far away. And it's not even like, I'm not even appealing to that to say it's impossible. I just want people to have a realistic understanding that like the more we're learning, the further back we're getting in understanding how life started naturalistically. And, I don't know if they'll ever fully admit it can't start, but there is a lot of like problems, compatibility issues, cross reactions, if it were to just form on its own that I've went over. So I went over like 10 to 20 issues so far. And they acknowledge them. It's just, it's rare for them to come out and say them this bluntly, like John Sutherland did, Dr. John Sutherland. Okay. Now, the next segment's going to be a positive case for intelligent design. Did anybody have any questions about the last segment while I just take a sip of water real quick? Or any comments or anything?
Yeah,
Great
I'd
job
like to
so far.
pretty much all of your, your critique is science can't explain it, therefore God. So, I mean, it's fun. You're critiquing these guys and laughing at them.
Do you have a
that they're-
question?
I haven't
She
said
asked
the word God in my
comment.
presentation yet. So whenever I say,
Do you have a question, Helios?
She asked me if I had a comment. I'm answering it.
it's fine.
Yeah, she did say comment or questions, I think, Briggs.
Yeah, it's okay. Just make it quick.
You have a problem with that or what?
Well,
Do you have a comment
I
or
was
question?
slightly annoyed myself. I did have slightly an annoyance because she literally predicted your comments on membranes with her paradox on the membrane presentation segment. So it's like I am a little bit like you're just saying the same thing essentially, like therefore God, and it's like nobody even said anyone's. We just pointed out there's a Schrodinger's paradox like right in our face, and however much we throw at it with the best scientific, you got to keep in mind, this is my comment. There are rooms and rooms and rooms of computers at NASA working for their interests all the time, and for
So
any major system like CERN or anybody who's studying these fields with money behind them for their very particular worldview. That's my comment. Like, there's something weird here where we're making up arsenic forms of life as some sort of like publishing, and then the independent researchers in this field, which are also very heavily funded, see, hey, that's fraud.
Yeah. Yep, they got called out for it. That's pretty funny, huh? I don't know if anybody else is ever calling out NASA, but I know the biologist did, so respect for that.
So can I
Okay.
still give my comment or?
Make your comment. Go ahead.
So yeah, you're just saying intervention is required. You did make that claim that there has to be something that intervenes. So can you show us that process or like the exact moment or which moments there is intervention to make all this happen?
I'm not claiming to like have a breakdown and have a full understanding of how life was. I don't think anybody can say, oh, definitively, God, yeah, he like formed the clay, and then whenever he commanded it to come to life, like all these processes just started happening just to do as he commanded. I mean, I can hypothesize, but I can't recreate it. So for me to sit up here and act like I know how God did it, I'm just showing you like the current state of research in the naturalistic explanation, where I haven't even mentioned God or any intelligent design yet so far. All I've done so far is help you understand where we're at naturalistically to understand, the origin of life. This is where we're at. This is what we got.
Yeah,
No,
so
I
we're at. Hold
I
on, bro. Let me.
recognize. Okay.
Can I?
Go ahead. Yeah.
We're at our infancy, but making critiques and not complaining, but kind of you're scoffing at it, laughing at it, at our infancy and knowledge. What good does that do? Yeah, it's good to highlight that, but yeah, that's what.
I'm pointing. It is funny. I mean, they'll even laugh. I mean, they even stated at their convention that they can't come to a. They were laughing at themselves. Look, the RNA world hypothesis, the worst theory of the early evolution of life, except for all the others. Are they not laughing at themselves? This title, bro. Like, it is kind of funny.
Okay,
The
it's funny that, science has more of an explanation than what the Bible does, though. Like, Bible can't even go into detail of all. Can I
We're
finish?
simply
Can I
pointing
finish?
out.
I
You've
was-
been talking a while now.
Wasn't done. Can I finish? So I was saying, science hasn't figured it all out. They've given more of an explanation than the claim that God did it and the Bible. You just don't. You can't let me finish, right?
I didn't bring up the Bible either. So can we stick to the topic here? Is naturalism sufficient to explain the origin of life? That is the topic. Do you have a comment on that? Can naturalism explain the origin of life? That's the topic. That's what I've went over so far. Is there a comment on that particular thing?
Yeah,
You really want me to go into that? Because that's going to take a long time.
Just present it.
Before he does, I would like to comment, and then he can go right into it because I'm also willing to hear it.
Yeah, of course.
Okay, all my comment to put forward is there is this idea that we can retract back to this thinking of science still sits as a baby in the crib, yet we declare as though our minds are still babies in the crib to children as though we are authorities on the matter 100%, as though there is no baby crib scientific matter going on here. That's the problem at hand. And since that's the current projection into classrooms, into soft, childish minds of little people, I'd say that's pretty disgusting of a claim to hold Helios as the worldview to then impose, considering we recognize it's simpler to tell the child it's not understood yet. Okay,
So
that's
you're
all.
Well stated.
I mean, agnosticism. Cool, got it. So don't assert a God to us then that God did it, and we'll
I
be
wish that
good.
they would be agnostic at the very. I wish they would be
clearly
honest.
Otherwise,
That would be
you
nice.
can't
If they
say-
said we don't know, that's huge progress. If they said we don't know how life, boom, I'm happy with that, dude. Totally happy with that. But they pause it as if they understand it, and that's the issue.
Even then, it's hard to be agnostic when you know there must be an intelligent agent which designs this.
Yeah, I mean, still, but if they even acknowledge like we don't know in a naturalistic state how this happened, I would be like super happy about that. But they don't want to do that. They don't tell the public that we don't know. They want to act like we know everything.
Under no other circumstances has complex and specific information ever arisen without an
I'm getting that.
agent.
That's the next segment of my.
Helios said he was going to break something down. And
Is the Bible sufficiently explained?
Talking about the Bible right now, Helios. That's the topic.
we're talking secular. This is a secular breakdown of just literal science that can be seen, tested, and from a purely secularized worldview. Like that's what it is.
Not bad at all then, and we're good.
Nothing biblical. I'm just going over science. Do you have a question about the science? Please, Helios, don't bring up anything off topic.
No, I'm good. Let's talk about.
Okay, let's talk about
I
it.
love it.
Okay, so just now we went over a lot of different paradoxes, their acknowledgement of, the lack of progress in the field. I think these paradoxes, I can send this to Riggs. Maybe he can post them so you can have these. Water, energy, UV, hydrothermal vent paradox, heat paradox, natural selection, time paradox, DNA protein paradox, tRNA paradox, Huygens paradox, membrane paradox, circular synthesis paradox, ribosome paradox, etc. There's a lot of issues here. Not even just like we don't know how it happened, like there's actual inconsistencies and compatibility issues at play. All right. So it's not like, oh, we don't know. It's like, whoa, there are serious problems here. It's not even like you can't get around it. Even a new theory, you're still going to have, no matter what you conceive of, you're still going to need RNA to come into contact with amino acids to make proteins to get this whole transcription translation system going. But if they cross react, how do you get over that? So I don't know how they're going to come up with an explanation to some of these things, and sometimes they just brush them off, move forward. But like we said just now, like we're still in the very infancy stage of origin of life, where we're just trying to find the prebiotic chemistry that would need to be required for these polymers to come about to form life. All right. So the next segment this is a completely different segment than the naturalistic worldview. This is looking at a positive case for intelligent design as an alternative. Not talking about any particular god or any particular theology or whatever. Just the the notion. And there was in a magazine I read the other day. There is one a title for this now, like the alternative theory for original life and for other things, where it's like an intelligent design is incorporated without positing any sort of God. And some people even, just go by saying intervention of some sort would have to start life in the universe. But anyway, so this is a positive case for an intelligent designer. Okay. So indicators of an intelligent designer in nature. All right. So we can see things that are intelligently designed, like a computer. Nobody in their reasonable mind would say that this computer formed on its own by natural processes. If we find a piece of machinery, an engine, we can assume that, this was composed by somebody. And we know what things that are intelligently designed look like because we actually intelligently design things ourselves. All right. So complex specific information is an indicator of an intelligent designer. To have organized, meaningful complexity. Also, irreducible complexity in the definition that all parts are essential for function. Not that, if you remove one part, it still works, therefore irreducible complexity debunked. No, that's not my definition of it. My definition is that you need multiple functioning parts for it to work. Okay, you have to have more than one.
Right now,
And
I
then
have to.
purposeful arrangement. What?
Are you invoking God?
What?
Are you invoking God here?
Where on this slide does it say God?
Intelligent designer?
Yes, I'm giving you indicators of an intelligent designer in nature.
You said
If
you weren't gonna. You said you weren't gonna invoke God.
No, maybe chickens.
Stop
Listen,
complaining, Emu.
stop. So for my last segment of my presentation, I went over just the original life as it stands on its face. This next segment is completely separate in that it's a positive case for an intelligent designer. So right now, I'm giving you attributes of things in nature that we have and can touch and can feel and see, that attributes of something that is intelligently designed. Okay, do you understand what I'm doing? So just give me one moment. I'm going to keep going through this list. All right, so things that have purposeful arrangement, right? Features designed for specific goals. Fine tuning, calibration, precise parameters, information rich features, detailed, intelligent associated information, specific complexity, non random patterns, matching specifications, adaptation of means to an end. So parts working together for a clear purpose, creation of new information, novel function information, all of these things being present would suggest intelligence was at play. So if we were to find, an engine on the side of the road, we can go through this checklist. Does it have purposeful arrangement? Does it need more than one part to function? Does it have complex specific information? Is it fine tuned? These are all things, attributes of things that we know and can design ourselves. These are attributes of something being intelligently designed. So anyway, these are key indicators whenever we're looking at stuff in nature to see, did this come about by happenstance, by random cosmic coincidence, or was this purposely, constructed? All right, so here's irreducible complexity in integrated systems and protein synthesis. Okay, so the systems of DNA, mRNA, and tRNA and ribosomes work together in a tightly integrated network. Each component is dependent on the others for its function, or else death or disease occurs. So you need to have these things codependent, integrated, working with one another. All right, and they're required for one another. All right, so here's a segment on tRNA, and Dan, me and him worked on these slides a while back, so he contributed these to my presentation. So if he still has a good recollection of them, which I believe he does, he's going to go over these for the next couple minutes.
Hello, everyone. My name's Dan. I'll talk to y'all about translation. Real quick, does anybody have any questions or like comments, kind of like where you're at on understanding translation and transcription?
I went over it a little bit earlier, but basically it's essential dogma of biology where you have that DNA, which goes to RNA, which goes to protein. So the transcription is from DNA to RNA, then from RNA to proteins is translation. But to get translation to occur, you need to have these this machinery already present. So he's going to go over the translation machinery and what's present to have translation occur. Yeah, I kind of did it back then.
Yeah. Like I know this wheel looks kind of confusing, but if you were to use like AAG, then that gets you to lysine. Like all proteins consist of these amino acids around the outside, and then the codon is like the letters that get you to that amino acid.
So
I don't-
three
Yeah.
nucleotides code for one amino acid. And remember, proteins are made up of amino acids. So we have the DNA and the RNA, and then in sets of three, these three nucleotides in a row on the RNA, that makes a codon, and that's going to code for a particular amino acid, which is going to then be brought and linked up to make a protein. Okay.
Yeah, cool. So that's a really good introduction right there. But the point of that is to kind of explain that there's two highly specific and complex languages that are happening all at once. And it consists of different spelling, the orthography, and the different units, morphology. And it has to be matched up like just perfectly. And if it doesn't work out, there's even a reversal that can correct itself, which is pretty cool. So there's 64 possible codons in mRNA, and it has to be matched up to the 20 amino acids. An important component of this is the transfer RNA. That's frequently overlooked in biology courses and stuff, but it plays a role in the ribosome. Let me actually here. This is a detailed model of a ribosome. It's what transfers mRNA into protein. I'll like show this first because it's kind of helpful. That like here's that mRNA that's coming from the nucleus, and then it's going to be matched up with a tRNA. And I'll come back to that. I just want to kind of set the picture a little bit. So the tRNA is an RNA molecule, which is essential for protein synthesis. And it really works like a translator because there's a specific tRNA that matches up with almost every pattern of the three nucleotides which made the codon, like Taylor was just mentioning. And so this specific tRNA right here has to be matched up with has to be activated first. Okay, it gets active. Like this little tRNA is going to become activated by binding to the amino acid, and then the tRNA is going to be bound into the ribosome to help grow the protein chain. So just like I said, it attaches to an amino acid, and then that tRNA matched up with its corresponding codon and amino acid will fit into the ribosome and add that specific amino acid into the protein. And I added this picture here because there's like different arms of even just these tRNAs. Like they're not super simple. In fact, there's the anti-codon arm right here that's going to bind to the codon like you've been seeing in the pictures. And there's a T and a D arm for like proper folding and function. All of this has to be like really perfect, or else it's not going to fit into the ribosome correctly.
So you already have.
and
Yeah.
what is the brain that is like controlling these very separate and specific functions? Like,
I
because
think-
these are just the raw components, but where's the brain that's controlling all this?
I kind of would ask a similar question to like my biochem teacher, be like, well, what's the cell like thinking here? Why is this happening? And she'd always say like, well, it's not thinking, it's just like doing. And I think that's like the kind of the beauty in this is that like you don't like shake around gravel and it makes like something new, like nothing builds itself out of nothing. I think that's kind of obvious.
Yeah,
And
you don't
then
have to even think about it. It's just happening.
Yeah.
It's like it's crazy. It's all working together like a city. Like I don't know, Riggs, but there's some life force that's
Yeah.
moving all these different
it's like functions built into the chemistry that has been arranged in such a way in which it, results in intelligence.
But it just getting it to go, like you can have everything there. Getting it to go is a whole nother issue. But anyway,
What's
Yeah, from what
intelligence, though?
I know that this, you're intelligent. If you were able to ask the question, you hit on mute, you said something, you're intelligent, and you're no accident. So I'm sorry if you think you are. This is a really important slide, but it just looks like a couple crazy pictures to y'all. But like I promise you, it's nuts. Okay, so this tRNA I've been talking about, like it being the translator, it has to bind the codon, binds amino acid. There's an enzyme right here, okay, that makes it bind the amino acid, amino acyl tRNA synthetase. A preformed protein, amino acyl tRNA synthetase, uses one unique enzyme to bind each correct amino acid onto the specific tRNA in order to make protein. Guess what? This is a protein which has to be built in the same way. This amino acyl tRNA has to be built by other tRNAs binding to it, building it using a ribosome. I think that's pretty incredible, right? Like how the chicken has to come out of the egg, right? But well, where's the original egg? Okay.
yeah, it's
So
like a chicken and the egg problem, huh?
even more than that, right? All that has to happen for the ribosome to even have its components. Now let's talk about this incredible machine here. So protein synthesis obviously happens at the ribosome. And the ribosome itself is made of two very large RNAs and over 50 preformed proteins. You got an A site. The A site is the start site, where the charged tRNA, the charge, we just talked about charging the tRNA, binds the correct mRNA codon, moving to the P site, where the amino acid, we can go here, like see where this is bonding, and then where this next amino acid binds onto this, that's where it happens in the A site. And then there's the E site, where the tRNA can like kind of rest and then be kicked out before the next art, before the whole thing shifts over.
Next tRNA
Kind of like
comes
you
in.
see happening here. This is
Yeah.
this is sort of like the amino acid's been transferred over, and then the next one gets kicked out of the E site, and which one was on A moves to P, and then that can keep happening over and over again with new ones binding in. But the important thing here is that you have 50 preformed proteins forming the ribosome, and if any one of them are missing, the ribosome does not function and does not make proteins.
So are you telling me you need 50 proteins to have one protein form?
That's correct. And then I threw this in here. I'm sorry, I think I might have misspelled something. But if you thought that was it, this is pretty much everything that I described, but really summarized down. Okay,
So here's the ribosomal components. The bacteria ribosome. Here's like the most simple type, right?
yeah, this is a bacteria.
And so the bacterial ribosome contains 54 proteins. And this is going over all the different components of
the translation.
ribosome.
So I
Yeah.
talked about initiation at the A site, elongation, and termination of the protein synthesis in the ribosome. And I
Yeah.
think Taylor wanted to keep showing y'all some awesome examples of
I think one of these might
intelligent.
be, yeah, like over here. Well, this is actually DNA translation.
Yeah,
But
this is
yeah, I think
translation.
that there is a schematic that I should add about that tRNA and its complexity. So just to like hone in on that amazing demonstration. Thank you so much, Dan. You got like so much complexity just for this one little machine that's required for one process of making one protein. So remember, the protein's what carries out all the function in the cell. Well, you have to have one of these you have to have this present to make one protein. And what this is look at how many components it has. You have the P site, the decoder center, the pre-accommodation tRNA, the EFTU, and you got the exit tunnel, the large subunit, peptidyltransferase center, E site, L1 stalk, and all of these have certain functions. And if you're missing those, you're not going to have anything work. So, and this is needed for the most basic needed for even the most basic functions of the first cell. So
are those each intelligent?
What's that?
Are those each intelligent?
I would say something's going on. I've heard about so much.
They're performing a purpose, right? Like if we're talking about engines, is the camshaft intelligent? Are the rocker arms intelligent? Are the valves timed correctly? I mean,
That's what I'm asking. Is the mechanic part that you're calling mechanical or machine-like, is it intelligent?
That would be
And
up to
Yeah.
so,
some.
if so, then your car motor is intelligent too.
Okay. Yeah,
I mean, the one who designed the car motor is intelligent, right? It has the function. You know, Rudolf Diesel did a great job designing a diesel engine for a purpose, right?
So only designer is intelligent, not the thing that's doing the stuff afterwards?
Well,
Okay,
it's
so the
receiving instructions from somewhere, right?
I'm sorry.
the motor is intelligent, but where did that intelligence come from? It obviously came from the one who designed it. So the designer of the motor is where the intelligence is based from.
Where did they get theirs?
Well, unless you want to fall into infinite regression, you're going to have some sort of prime mover and creator.
Okay, well, what is it? What's intelligence?
What is intelligence? That's not the purpose
Yeah.
of this.
That's
That's
sort of
not the. That's
semantics,
not
right?
I don't know.
Because
I feel like I'm looking at it. I'm looking at something that, to me, like what I see in front of me is a straight declaration of some miraculous event. That's all I can put forward.
Yeah.
You can ascribe to different miraculous events for your own,
So
but
those
there's something here which says, without intervention, without literal miraculous intervention to synthesize this first unit, that there's no instruction set to begin the instructions. There's no mechanism. There's no vessel even there to initiate the instruction set. There's nobody at the throttle to press the throttle. There's nobody there to turn the steering wheel. You know,
Can you
there's no.
tell instruction from intelligence or learning from uploading, right? Can you tell the difference from an upload to
Yes,
learning
because
in a
if
computer?
if we put a brick on the accelerator of the
Can you listen
car
to the
and we
question?
put a
Can it,
rope, that's what I'm saying. It's like, yes, you'll see the pattern of no intelligence behind the wheel
so,
when there is no intelligence behind the wheel.
so in
But
a
when you put
computer, you can tell that it didn't learn anything, that it got uploaded information, and you could tell a human has learned information and not uploaded. How do you tell the difference?
first, I think you'll have to clarify your initial analogy here.
Uploaded information, right? Like you download or upload
It's
something
storing.
into
Yeah, it's
something,
storing.
right? Can you tell the difference between that, right?
Are you saying, can you tell the difference between memory with input from
No,
a outside exterior source into
no,
a bank? Because that's what I'm hearing.
no.
I'm
I
hearing a
said,
process that requires an intelligent mover behind the upload of the file.
can you
I'm
tell
just
the
boiling
difference
your analogy
between
down
which
to its
is,
principal. I'm sorry, I'm just boiling down your analogy to its principal beginning, where I have to start for your analogy to be correct.
well, you shouldn't do that because you're wrong. So
Why
what I'm
if I
asking
boil it,
is,
distill your
what
argument down
can
to
you
the
hear?
beginning?
Can you hear me? What I'm
Yes.
asking is, right? Can you tell the difference whether a computer has uploaded the information or learned it?
I think that a computer innately has a designer because computers don't form on their own. So whatever's uploaded the information is obviously a source of intelligence.
Yeah, we
So
showed like a
can you tell whether you have uploaded information or been downloaded information or whether it's learned?
I just want to say that like we're way more complex than like uploading and downloading information. Like
That's
memory
what
is
we're saying.
composed of perception, integration, and recall. It's like each one of those are just beautiful designed circuits across the brain that like require so many different things to work right for you
Where's the memory at?
Well, that's a big question that neurologists are still trying to uncover is how do we store and recall memories? I mean, it's all miraculous. All of this is miraculous. This stuff does not happen on its own. That's my whole basis that I'm trying to lay down here. I don't know if
So
you
it's
caught.
miraculous.
We're gonna have a Q&A at the end of life forming on its own.
we only got
We're gonna
4
have a
slides.
Q&A at the end, but let me get through this and we can talk as long as you guys want.
And
Okay.
we can go back too. Let's
Okay.
just finish up the slide.
Yeah, but I just want to say with this one real quick. This is really amazing because there's a language system, and Daniel kind of mentioned this. I just
Yeah,
wanna
I mean,
point out. There's a language system within the DNA, And there's also a language within the proteins, because you have the amino acids, which make up the proteins, but you have the nucleotides, which make up a DNA. And remember, 3 nucleotides code for 1 amino acid. So you literally, not only do all these components need to be there for this tRNA to function, but this is a whole translating system already embedded into the cell. So you have 2 languages and a translator. And, you know, not even like thinking about the components, like how did this translation system and the information translation come about? That's a huge question. And earlier in the paper that I pointed out, they even said it's amazing to think how this translation system even came about. Like, and that was in a modern day abiogenesis paper on Origin of Life. But yeah, to like not only have these components come together, but we haven't even got to like information side of it. Like now we have like some translator who's able to accurately decode the nucleotides and make them form a protein. So yeah, I just want people to get like the, you know, how amazing this process is that's actually occurring within, you know, any cell reproduction. So just think about that. But yeah. Okay, so and maybe later I can go back to that quote in the paper talking about the tRNA. They acknowledge how crazy it is too themselves. All right, so here's indication of design in cellular machinery. All right, so right here is the flagellum, and you can see it has like a rotary motor within the flagellum. And right here in the bottom left is actually DNA replication. And you have basically all these different working components that'll come. Do you have anything to comment on this one with the
No,
loop?
not really.
What about there's like a lagging and a leading strand because they can't like replicate it backwards, so they have to go down this lagging strand.
That's the difference between this one going over here and then like this one making a loop. But
Yeah, because they can't replicate it backwards,
I need
but
a
they're
bunch of
going down
pictures to show
because
you.
it's a helix, right? And so they're like they're like bonded to another.
And the 2 sides of the helix go in opposite directions, and this machine only goes like one way, so the other one way is going backwards. You know,
So you'll
basically.
see one, so that they'll form like a loop to get around that.
Yeah.
And just go do that one loop at a time. That's why you see that loop coming off. But
You got a
apparently
bunch-
this DNA polymerase is spinning and working faster than a jet engine at almost 100% efficiency. So
Yeah.
its machinery makes our machinery that we create look like child's play, the machinery within the cell.
There's a bunch of these little things that are like moving around like this, like pivoting and grabbing something and rolling. It's like
They all have a
like,
purpose, yeah.
this right here is like the stator of like a turbine. Essentially, it's like driven by protons in the mitochondrial membrane, and so it rotates. It pushes this right here, which rotates these arms so that it can click into these different positions and make ATP, which is the energy of the cell.
Yeah, ATP synthase.
This is even still like a mystery. Like you can learn about this in biochemistry, but I've read many papers that basically explain we don't really know how this thing works.
Yeah, it's amazing. I mean, you can look at them on a fluorescent microscope, like spinning and the moving components working, but like, man, this process right here is absolutely incredible. And then the kinesin walking down. So this is how things are delivered within the cell. So you have a kinesin, which is a transport protein. So it's moving things down the microtubule. It's carrying some package, you know, to different parts of the cell. So that's how and it just looks like it's so alive and walking. Isn't it so cute? The kinesins, yeah. But anyway, that's how things get moved around the cell. All right, and so here's the similarities in ATP synthase, which we just went over, and man-made motors. So here's actually pictures of the ATP synthase that they were able to take. And you can see over here, what is this? The rotors. And I don't remember what these are exactly pointing to, but they match up with like machines and motors that we have now. So ATP synthase and man-made motors share key similarities. Both utilize rotational mechanisms with a central shaft rotating within a stationary housing, resembling the rotor and stator design in electric motors. They generate torque to perform work and are modular in structure and adaptable. ATP synthase can operate in reverse as well. Additionally, they convert energy with remarkable efficiency of almost 100%. Did you want to say anything on this about the housing?
I mean, they honestly are little electric motors, just not like conventional ones we think of. Like it's truly a motor performing work
Yeah.
using electrical potential.
Yeah, same
Pretty
thing as a
nuts.
car. And so this was interesting because we actually created motors to do work. We said, oh, here's the most efficient way to do this thing to get, tires to move, for example. This is the most efficient way. And then later we look inside the cell and we find the same thing that we designed. It's like, oh, cool. We made this too. We found out this is the most efficient way. It's funny how we find it in the cell. Could it be an accident or, whatever? Anyway, it just looks very it resembles what we've designed as well, and it needs multiple working parts in order to function. It almost seemed like it had an end in mind whenever it was being created, like not by happenstance. Because remember, why would it keep evolving and having natural selection to form this thing if there's no end in mind? They don't know what it's going to. It's just a set of, random events. And, why would natural selection even preference to have this thing start forming if it doesn't do anything? Because unless you have all these different components, nothing's going to happen. It's not going to be able to do its work. All right, so now moving on, I'm approaching the end here, so I know I'm going to wrap it up soon. But intelligent design and DNA, this one's really important. If you're just trying to make a positive case for intelligent design, the DNA code is a very good one. So complex specific information. DNA's code is highly complex and specifically ordered, information being a message or instruction within meaning. So some people ask, what does information mean? So information is a message or instruction with meaning. All right, so information-rich features. DNA efficiently stores and transmits detailed genetic information. It has specified complexity. Base pairs are complex and non-random, matching specifications for cellular functions. They have a purposeful arrangement. The structure is designed for sorting, storing, and transmitting genetic information. It has irreducible complexity in that the DNA replication process itself is irreducibly complex, like we saw on this little motor right here. It has multiple parts needed for even, for any DNA to be replicated. All right. And then the last one is fine tuning. So it has precise nucleotide arrangements in DNA sequences, which produce functional proteins. So it's meaningful. The code in DNA is not just like meaningless. So you can see rearranging patterns in nature, things that repeat, like a crystalline structure, for example, will have repeating patterns, but it doesn't encode for anything. It has no meaning to it. So it's just like, something repeating, but it can be complex, but it's not specific. So while a crystal can be a repeating pattern of complex stuff, like it doesn't have any specificity to it. Like it's not coding for anything. You know what I mean? So complex and specific information. All right, here are some quotes from people, huh?
You still have this slide?
Yeah, I think it's at the end.
Okay.
So Bill Gates, he said DNA is like computer program, but far, far more advanced than any software ever created. Richard Dawkins says the machine code of genes is uncannily computer-like. Francis Collins says DNA is something like the hard drive on your computer containing programming. And Craig Venter said life, and he's a biotech guru. Life is a DNA software system containing digital information or a digital code. All right, and here's the sources underneath it. Dan, here's your picture if you want to say anything about it.
Yeah, I love this picture on the top right.
So wait,
So-
read this real quick.
Yeah, so circuitry, informational hierarchy, and integrated systems in computer chips and cells. So just like intelligent humans, I guess, designed integrated circuits that are precisely engineered with billions of transistors. The cells have circuits composed of integrated molecular components performing complex information processes. And I just love this picture right here. I know it seems like really overwhelming, but this is basically human biology, biochemistry. Like this little line right here is glycolysis, you may have heard of, and like this right here is a citric acid cycle.
Yeah.
Yeah, and like energy for the cell. If you see here, I know it's very small, but each one of these little blue labels, they're all over, all these little blue labels. Well, guess what? Each one of those is another enzyme that it's made just like a protein, made through translation, like we talked about earlier.
It is a protein.
Yeah. I just think it's incredible that all this stuff is all it has to be making energy in order to make these enzymes. And then the enzymes have to be present to be making the energy and making amino acids. Yeah. Yeah.
That's so funny. What is it here in the middle? Do you know what's
Yeah,
happening?
this is citric acid cycle here.
Okay.
And like, here's how you make fatty acids. here's how you make nucleotides. So everything in DNA and RNA is
And they'll
made down-
overlap. Yeah. It says DNA and RNA synthesis. So everything kind of overlaps. Like one product from a reaction will be used for another. So it's all needed to work together in one, just kind of like a computer chip. You'll have multiple parts and, it'll have information encoded, go through multiple different circuits. So, the mechanisms within a cell are kind of like a computer drive where they all have their own specific function. And, we see that computers can, try to simulate some of the work that life can do, and they require the same components as life. But, we both know that these are, we can all acknowledge that this is intelligently designed, and, maybe try to think about, can a computer chip come about by happenstance? It might be even more difficult for a computer chip than cell, but that's another argument.
And if you were like an electrical engineer wondering, are there like and or and nor gates? Yeah, there are like and or nor gates in biology. Just they're not super like clearly drawn out here. And those gates are what like make up a circuit. Just wanted to make
And
that.
what do those do, the gates?
It'd be like and it has to be both or one or the other or a nor gate.
Yeah.
One, right? So like,
Okay.
um,
Yeah, like in different like what is it called? Signaling molecules. It could be like, are both of these hormones here, or only one of them here,
Yeah.
or neither of them here? And then that'll drive a different reaction. So yeah, it is a lot like a computer. The cell, like a computer chip in a way. So we have a multi-layered computing architecture overview. So somebody actually wrote a paper on this. So computational architecture of the cell, evidence of top-down design. So here's a quote from it. The complexity of the cell and its corresponding analogies to contemporary computing systems run deeper than simply DNA. The cell as a whole has also been viewed as a computational machine similar to multiprocessing clusters with a combination of centralized instruction sets and distributed computing elements. So I'm not like a tech person, but he's saying the entire cell basically is like a computer. It has computational architecture. And so what this would indicate is like a top-down design where you build it top down with an end in mind. You have all these functioning parts that need one another. So it makes more sense that the cell came about in a top-down fashion rather than a bottom-up fashion. As we see, as we saw with the beginning of the presentation, we see you need multiple parts. We see these parts do not, like in the environment, come together and form anything useful. If anything, they're destructive, and the environmental conditions to form them are incompatible. So top-down design makes much more sense. And it says the channels of information transfer and transformation in the cell have direct analogies in communication theory and computational algorithms. So there's a lot of overlap here between the cell and computer architecture. And they have examples here, but I don't have to go through them all. Memory bank is like the chromosomes. You have the drive formatting and the histones, software algorithms and gene expression. Yeah. Interprocess signals with pathway crosstalk in cells. So there's a lot of overlaps, and it gets very technical, but oops, let me go back one. I'm almost finished. So, is abiogenesis scientifically plausible? Question mark. A scientific explanation, whatever it may be, any scientific explanation must be coherent and provide evidence to whatever phenomena it's trying to explain. Okay? Abiogenesis lacks this. It doesn't have a coherent theory, and it doesn't have evidence for what it's trying to explain. They can't recreate these things, much less from inorganic matter in a truly prebiotic condition that would, without heavy intervention. They're not able to replicate this process that they're positing is real. So, any hypothesis or any explanation scientifically needs to be coherent and have evidence, but it just lacks it. And if you say maybe someday we'll figure it out, I mean, I get it. You're just appealing to the future, though, where someone claims their argument or belief will be proven true at some point, some unspecified point in the future. Ironically, though, the more that we learn about the cell, the more we're seeing, like in this last slide, look how much we're learning is required, like for even basic cells to function. We had no clue that all this was necessary whenever we came up with this theory. Whenever we said abiogenesis is plausible, they thought I read some quotes from that time period. They literally thought it was balls of jelly, with protoplasm and a nucleus, and that's all. But yeah, now we're learning much more about it. Is intelligent design a proper alternative, question mark? So Occam's razor prefers explanations with fewer abstract postulates. So intelligent design does have an abstract postulate, which is an intelligent designer. But Occam's razor, I believe, would be on the side of intelligent designer because it prefers it has less abstract postulates. You're not having to claim that a meteorite struck the earth and it got hit by lightning, then that went to a tide pool and they had different microenvironments and some of them got struck by UV light, maybe struck again by lightning. I don't know. It's just craziness. You have to go through make so many postulates for even the most basic molecules, and it's just wild. All right, so intelligent design is not a God of the gaps argument, and here's why. It's not based on what we do not know. It is based on what we do know. So, for example, we do know where complex specific information comes from. We do know the attributes of something that is intelligently designed. We do know that the cell carries these attributes. So, for example, informational hierarchy, integrated systems. We know that these things are present, and we know that these things in nature come from intelligent designers. That's just what we do know, okay? And we do also know that in nature, we're not seeing where life is popping into existence, even in their best chemical soup they can give. We know these things. So whenever we say intelligent design is an alternative, that's not a God of the gaps based on what we don't know. It's a God of it's literally just going off of what we do know. It's not a God of the gaps. It's just putting together knowledge we've learned about the cell to make a conclusion. All right. Prebiotic experiments for origin of life research support the need for intelligence. So, for example, heavy planning, heavy intervention, and even the most basic experiments, like we saw with the Miller-Urey experiment, they put specific amounts of these types of gas. They literally did evaporation. They struck them with lightning. They condensated them. Then what did they get? They got a mixture of tar, and then they basically isolated the 2% amino acids and acted like that could be usable for the cell, even though they were not even usable. They're 2% from their tar because it was heterochiral. But in this experiment, we saw heavy planning, and this one's actually good on that side. There's even the modern day experiments have much more intervention and planning and regulations, but heavy planning, heavy intervention. For example, they'll isolate the products and then move on to the next step with its different conditions and pHs. So heavy intervention we're seeing is required for even the most basic components of the cell. And so here was Dr. George Wald, 1976 Nobel Prize. So intelligent design isn't rejected on scientific standards, but philosophical ones. So like personal incredulity, which is a fallacy when someone dismisses a claim or argument simply because they find it too difficult to understand or believe. So this is a quote from him, only one has to contemplate the magnitude of this task to concede that a spontaneous generation of a living organism is impossible. Yet here we are as a result, I believe, of spontaneous generation. Most modern biologists, having reviewed with satisfaction the downfall of the spontaneous generation hypothesis, yet unwilling to accept the alternative belief in special creation, are left with nothing. I think a scientist has no choice but to approach an original life through a hypothesis of spontaneous generation. So some people have actually misquoted him, and I learned this recently, so I changed this quote to what it actually said. But what he's actually saying is, even though there's a lot of evidence against spontaneous generation, there's no other option. He's not going to accept creationism. But he's still going to continue looking through the lens of spontaneous generation because he says he has no choice. Okay? This is whenever you have dogmatism, you're not going to accept the other side, even though there's a multitude of evidence that this is not reasonable. Okay, so this is funny. Like, the task to concede that spontaneous generation of a living organism is impossible. Yet here we are, and he still believes in spontaneous generation. Even though he just said the task to concede that spontaneous generation of a living organism is impossible. So are these people not saying that they're willing to believe something even though they know it's impossible? Like, what? That is incredulity, and that's fallacious. You can't just say, oh well, I don't believe in creationism, so I'm just going to keep thinking it can happen on its own by accident. Like, that's not how things should go. It's not, intelligent design is not rejected on scientific standards, but merely philosophical ones. All right. And I am open for any questions. Any questions, corrections, or rebuttals? I probably will add some more to this presentation, but that's what I have for today. And thank you guys for listening. Yeah, it kind of had an abrupt ending. I'm sorry, but
That's okay.
yep.
We're thinking. No, I think that this guy right here that you're looking at, that's a very interesting approach. It reminds me of the Copernican principle quite a bit, where it's
Yeah.
we will accept it only because we want it to be, and that's what I found also in my research. But go ahead. I mean, this is yours. Yeah, this
No,
was great. I'm sorry I wasn't here completely on time.
good. But yeah, this is a really a good way to end it though, because like with everything that I've laid out, like we see that there is no real naturalistic explanation. If anything, it's completely contradictory. That with full of paradoxes, full of incompatibilities. They even acknowledge RNA world. That's like the leading hypothesis. It's the best. It's the worst one except for all others. So none of them are good. There's no best hypothesis. They're all bad. They all fall apart. But they're not going to accept the contrary, which is intelligent design. Like he just stated, spontaneous generation. He made that statement after it was disproven that organisms can just pop out of existence out of chemical soups. That was disproven. But he says he's not willing to accept the contrary. So all that he's going to fall on is spontaneous generation, even though it's been disproven. Like, because they're not going to accept the other alternatives, and that's just where we're at. Even though there's a load of evidence for intelligent design within the cell, they're not going to accept it for philosophical standards like in the mainstream. And I guess I get it, but it's just upsetting. And at the very least, I would like them to tell the general public that we don't know. That's a perfectly acceptable answer. We don't know. Stop acting like we do know and that we have some theory that can explain it, because we just don't.
They're just scared, just like everybody else in here who doesn't want to accept it.
Yeah, they
I don't
just don't
think
want to
it's fair.
accept
No,
Show us.
this is directed manipulation. I mean, this is what you do. Like if you tell somebody something that you know is not correct, as though it is the most correct thing to hold dearly in your life, and you do so, then there's something behind that. That's not benevolence. That there is deep-rooted malevolence in that. I do not believe that anybody in good conscience can promote such a campaign as wide as the abiogenesis campaign.
So show us your theory then. Show us your alternative
Thank you,
explanation.
Taylor, for showing my theory, but it's your theory and the theory of the entirety of many of the field who actually investigate this topic.
Yeah, just
Yeah,
like
so show us
intelligent.
God then. So show
So
us
they
God then.
Okay, listen, just like the origin of the universe. They say, "Oh, well, we assume that the universe came from a big bang." They can't show us that big bang. They're going to take the evidence and say, "Okay, we can see the universe expanding, that's shaky or whatever." But anyway, assuming that that is true, if the universe is expanding, they just put it in reverse and they say, "Oh, well, it must have started with a bang." Can they show it? No. Can they explain how it happened or why? No. But they're just left with that, right? And so I think when we're looking at cell, what we see is that it doesn't form on its own. We do see key indicators of intelligent design within it. So I think that we should go with the conclusion it is at least plausible, and probably the most plausible explanation, that it was intelligently designed. Now, how it was intelligently designed, if you're asking me, show me how God did it, show me him doing that. I don't ask you to show me the Big Bang. We're just logically deducing that based on what we can observe. And we can
Can
observe
we
indication of design lack of naturalistic explanation.
can we see the little
You don't
things
have
that are
evidence that God
cartoons?
did it?
Can we what kind of camera do we put inside the body to see the little cartoon things doing their thing? The little
I'm glad that y'all asked, because I think that a lot of people missed some of these slides at the beginning. Some of these were pictures taken by me of living organisms. You can actually observe the processes going on within the living organisms. Here's DNA replication. Here's a white blood cell getting bacteria. Here's neuron formation. Here's you know, some diatoms. If you get a microscope and look at pond water, you can see. And nowadays, with good microscopes, you can actually see the components moving in a living cell, like in this protist right here. You can watch the living components as they move. And this is some videos I've taken of the microtubules growing in a live cell, not fixed. I'm just literally looking at it under the microscope, and you can see these processes occurring. So it's not all, you know, these animations.
What's a
All
cell?
you showed us, Taylor, hold on, D-Mug. All you showed us, Taylor, is natural bioavailability, but you haven't showed us any supernatural existence. So do you want to show us that?
I think
I'm
all of
seeing
it.
it everywhere. I disagree fundamentally. I have to speak up. That seems like such a just not a true statement. I've seen it throughout this entire thing. We're just going to agree to disagree on that between you and me.
You, and a fair perspective that has to be met in this presentation is that this is not a presentation for proving a specific deity. This is simply the observation of from the microcosmic of showing intelligent design simply based on its function and its composition. Like that is what's being shown. So
Yeah.
There's
like all
claims
the
being made that that there's supernatural intervention. So where is that?
I'm
simply
saying that
that
some
it's the
intervention.
difference between looking at some mud compared to looking at an engine. Like you can see the design that's implemented in what you're seeing. That is the
I know,
argument
but
being
your Bible claims that it took dirt from the earth and
this
God
the
supernatural. Hold on, bro. Can I finish? I let you guys go. It's on, man. There's
Don't talk
claims in your.
something that wasn't in the
Can I go?
presentation. Listen, I did not talk about the Bible the entire time. The only thing I invoked was intervention and intelligent design and gave the evidence within the cell for it. Okay, so don't bring up the Bible or anything else that wasn't in my presentation, please.
You can't really blame him though. He's so scared of the Bible, he can't even think of anything.
I know. Well, don't think about the Bible. We're not talking about the Bible. We're talking about
Yeah.
intelligent design.
We're not condemning you or nothing. We're not about to stone you or anything. It's okay.
Just look at the content in the presentation and respond to it, please. Is there anything within the presentation you want to respond to? Because nowhere did I mention any specific gods or any theologies or anything. I'm just going over the science. Did you kick him, Riggs? Is that why
Well, it
he's not
looks like
talking?
he's muted. He's been muted, it looks like.
Okay, just don't bring up, like, I'm more than happy to talk about God and Bible with you, but that's just not what's in this presentation, Helios.
Do you think bringing up intelligent design is scientific?
Yeah. It's going off of what we do know, what we can see,
Do
what's
you think it's
repeatable.
Do you think it's falsifiable?
Yeah, definitely.
How?
If we were to see, for example, there was a big jab at the intelligent design community with what they call junk DNA. They said, oh, in evolution, we would expect to see a bunch of junk if it just accidentally evolved. You'd have a bunch of leftover junk that wasn't useful. And guess what? They spouted out left and right this junk DNA. But guess what? Actually, we learned that this junk DNA is not actually junk. It's just non-coding regions. And it's required for the proper areas that are coded to have a similar activation site. So the activator can go bind to the proteins that are needed for a specific function. They can be in the same location when wrapped around the histone. So we found out that this is not actually junk DNA, which actually is a prediction by intelligent design that there wouldn't be such a thing. And the more we learn about the cell and DNA, we see that that is not a thing. So yeah, I mean, junk DNA, for example, are non-usable parts, like parts that are just left over from evolution and, you know, whatever, that don't have any function. Yeah, that would be some evidence against it. And to really falsify it, you need to show chemicals forming life in a lab. You need to put the chemicals together, make them do the thing that you claim happened, or at least posit some sort of working model as to how this could have happened, at least some conceivable notion at the very least. But we don't have a conceivable notion, nor can we replicate it whatsoever. So maybe if you could
So
actually
if we do it in the future, then that would falsify the.
yeah, go ahead and show
intelligent
chemical
design.
forming life. I'm open to
No,
yeah.
I'm asking you if we can show it in the future, then that would that would falsify intelligent
She
design.
did say you can appeal to the future. She did say that earlier.
Yeah, don't.
No,
I would.
she actually laughed when she said that, but maybe she was being serious. I don't know.
Well, she said it. She said it though, but I recognize at least the ludicrousness of just appealing to let's just keep kicking the can down the road because we might actually learn more if we explore this side of the
Yep, that's
scientific
how
field.
science
We might
works.
actually we might get further along scientifically on this side of the field. And were it to continue to show that we get better understanding of the said field, a higher resolution, and it doesn't seem to break down, then it seems like if you're getting into higher resolutions, that somehow you're getting closer to reality. That's what the initial concept of people's eyes, at least, have been, is that the better pictures we take, the better instrumentation we design, the better model we draw, that resolution increase gives us truer understanding of the world. And so at this present moment, it appears as though the current mainstream field is rejecting the area of highest resolution to then propagate incredibly not even just poor resolution, but counterfeit resolution, counterfeit projection, counterfeit fields of travel.
Well,
Because
see,
inside
Taylor,
sperm,
how do your professors take this presentation?
I've only done this presentation on Discord, but when I've talked to my professors about the origin of life, they totally agree. A lot of people agree that this stuff doesn't happen on its own. Like, there is very shaky evidence at best for the abiogenesis hypothesis, and they'll agree with that basic notion. A lot of researchers who are actually looking into the field are real about where we're at in the field.
is that stuff inside of a sperm or an egg? All that stuff, the heart and the brain and stuff, like which one is that in,
So
the sperm or the egg?
that's after cell specialization. The heart, but the information for it's in the sperm and the egg. Where do you think it comes from?
Well, I don't know. I'm saying in the sperm, do you see all that magical, crazy cartoon stuff you see in the blood and the cells and all the other stuff?
Yeah, no, actually there is in the sperm. This on this slide right here. Let me share my screen again. This is from a sperm. Let's see, share screen and then share entire screen. Here you go. Here's the flagellum right here. This is how the sperm moves. So this is of a sperm cell that you can see under a microscope. And there's also another slide looking at the sperm. If you slice the flagellum, this is what it looks like. Hold on. Top left picture. Yeah, all these organelles are also in the egg too. Yeah, all this is present within like the egg cell or the first cell for life. Right here. I think it's back here. Right here. Yeah. So this is like all these different subcomponents that we can see under the microscope are present in the egg, like in the first cell.
My eyes feel different.
The eyes are amazing. Anyway, was there any other questions or feedback or criticisms or whatever?
No, it was very technical, and I have a lot to learn.
Yeah, well, I try to break it down as simple as I can. But it's really the complexity that says it all, and just the paradoxes, like the fact that it needs water but can't have water, the compatibility issues, it needs energy, but energy is destructive without a form to use it. UV, it needs UV, but UV will destroy it over time. It needs heat, but heat will destroy it over time. So these are just basic concepts that you can take away from the presentation.
Taylor, can you remind us like your major or what your focus was in school?
Yeah, it still is cell biology.
Cell biology? Okay, thank you.
So do you believe that that because of genetics we're related to monkeys or no?
Well, what's your best evidence for that?
Or genetics?
Like, what about them? Like the Y chromosome being much different than the monkeys?
No, that our DNA is so close a match to them.
Well, it's so close and matched to all living organisms because you have something called housekeeping genes that's required for all life. So it's like, don't reinvent the wheel, right? If it
Yeah,
works,
no, I'm talking about how we're different from other animals, but we're most closely to apes. So I'm sure you studied that in biology. So I'm just curious what your thoughts are on that.
well, it depends on how you define close, because if you just use like the DNA that's being transcribed and translated, some people say that we're only 84% similar, but then again, we have a high similarity with all organisms because those basic genes that are needed to function in any life form.
And we were just watching a video about this. I haven't read much about it yet, but chromosome numbers. There's actually wild variation in chromosome numbers between humans and chimpanzees or gorillas. And what they were explaining is that we're actually more closely related to gorillas than chimpanzees, which goes against everything they've been saying about chimpanzees. And then what they were really asking was like they made it very clear. I would need to go over it again. I'm sorry, I don't know the details yet, but they made it very clear that we are not descendants from chimpanzees. And you'll hear people say, "Oh, we're so closely related to a banana," and all this. Well, it's like it's just ridiculous. Like as Taylor explained, and like what you saw in that biochemistry slide of all those enzymes that have to be functioning, like they work pretty much the same way throughout all life.
Yeah.
Well, how do you explain endogenous retroviruses though? Have you thought about that?
Do you think that ERVs have no function?
I think they have a function. They can be given function, very weak function in a lab.
Yeah. So if we find that ERVs have functions in the cell, would we assume that's just some leftover DNA strand from a virus that got introduced a while back?
What do you mean by assume? Like it actually can perform the functions that we know take place within a viral cell. Am I wrong?
Wait,
No.
you mean just jumping genes, right? ERV is just is a transposon.
So the ERVs is like, yeah, these sequences of DNA in the cell that they say are leftover remnants from viruses that got introduced a while back, but actually we're finding the ERVs have functions in the cell. Let me pull up what those functions are, but essentially, with that, it is yeah, it's showing that they're actually were put there for a reason.
Not all ERVs may have originated as insertion by a retrovirus, but that some may have been the source for the genetic information in the retroviruses they resemble. From my understanding, this is just a jumping gene, right? Like a transposon. This is a part of our like endogenous immune system against viruses.
Yeah, so ERVs, from what I remember, their sequences actually fight against viral infections. So it seems
Right.
like they're placed there for a purpose rather than just leftover remnants of DNA from viral infections in the past. And that's what I think about them.
Anybody want to prove viruses or DNA for that matter? Or we have to take that as objective fact?
Did you see my picture of DNA?
I mean, I've
How do you
ruptured cells
Yeah.
and like isolated DNA and then like run PCR tests on them and stuff. I mean, I don't know. It seems kind of believable.
I've
Do you
inserted
think that
plasma
people
codes for different
PCR
functions.
tests are fraudulent.
Okay,
Barry
listen.
Mullis
people
was
okay,
He said he
can be.
I'll wait.
Okay, so I've also worked with plasmids where you basically insert a strand of DNA that has a certain code that has a certain function. Well, you can make what's called a mutant. You can order the plasmid, you can insert into DNA and see that function be carried out or added. And in my strain I was working with, it was to make fluorescent microtubules, which I actually work with now. So there's something
So if
holding
you,
information to pass from your parents to you, like what's holding that genetic information in the cell.
so flu
'Cause you guys,
viruses are all placebo.
I never said anything about viruses. I'm talking about DNA
I know you
spread.
didn't. I was talking to the phase. I'm sorry.
I don't know what he's gonna say.
I think we had this conversation though shortly, just before Christmas sometime, Taylor, about flu viruses.
Just prove viruses. That's all.
I meant flu vaccines, obviously. I'm saying viruses, excuse me. Well, my question was, are flu vaccines, are they all placebo? Or do they serve a purpose? Are they meant to
Or
help
are they
people? Do they help
poison?
people?
They're
Do they
probably
poison
poison. Yeah.
people? Who knows? I mean,
Yeah.
they have an effect. We see they have an effect, but do they help people? Is what you're saying. We're saying, do they poison people? I'm not. I mean, I'm just positing there's other alternatives than just the word help.
Do any of y'all look into exosomes? I know that sometimes I hear people say that all viruses are just exosomes.
Yeah, I've heard that before, but I'm more focused on DNA and it being real because the DNA code really does give evidence for intelligent design.
What happens if you can realize that there's no proof for DNA?
Then it's
Does
just some
the whole
other magical thing that's making mRNA. I mean, what else is it? Do you have a postulate for what could replace DNA?
I have
Well,
no
it's not
proof for
even.
DNA.
Yeah, it's not even the idea of replacing DNA because the problem with the viruses is that no virus has ever been isolated in a scientific research. Period. DNA has been isolated, like to show its individualistic nature.
I don't know. I found electron microscopy of SARS. If you want to see it,
No,
Taylor, what are your
there's
thoughts on vaccines?
Is it like a cartoon or something?
there's no
no, you could
isolation
see it.
of a virus.
Yeah,
this
it
the
is.
qu-
It
is this
comes
the
down to a
question and
really
answer?
shitty graphic.
Can I
Yep.
can I ask a technical question just to you, Taylor? Is this the Q&A for people interacting with you, or what are we doing? Because I feel like maybe the chat, maybe I'm misunderstanding, maybe the chat's misunderstanding. It's like an open forum type deal. I just want to be respectful.
Yeah, thank you.
I'm just saying using
Taylor,
one
Taylor,
dogmatic
you're
axiom
muted.
to kind of prove or affirm another dogmatic axiom is a total waste of time.
Yeah, but I'm
But it's cool. Like that, some people just believe stuff. So I mean, can't really argue with that.
Yeah, but
My-
not
my-
only do they believe it, they propagate it.
I see. So it was mentioned there's electron microscope pictures of viruses, SARS particularly, and I suppose what I'm curious about is what functions are seeable under this electronic microscope, because what I'm seeing in the DNA is extreme movement, extreme complex, what appears to be instruction sets running, very similar to like a computer, whereas in this picture it sounds like a picture, like not even movement. Of SARS-CoV, it could, I mean, again, like I'm saying, like, do these help people? Do they even perform functions other? Because I know I can drink arsenic and it'll kill me, like a couple hours later. So I mean, like, what is the thing? The arsenic doesn't appear to move to me under the I mean, does arsenic move under the microscope all by itself? I don't know. I haven't looked at that. But
So I don't
to
argue
me,
for
like what
virus.
I'm hearing is just
I'm sorry, go ahead.
All I'm saying is like I'm just seeing that yeah, we can take pictures of things, but do they perform functions in these pictures the way that DNA is obviously performing these immense functions?
These pictures are taken. I'm sorry, I'm going to cut you off. I was just wondering if you knew how these pictures are taken because they're not videos like scanning electron microscopes. Everything has to be like frozen, sprayed with gold. You're literally capturing pictures of reflected electrons. A lot of the problem comes down to like optical resolution. Like when things are so small, you can have a
Yes.
resolution which is smaller than like the wavelength of light or whatever. And so you need to use other like mechanisms to view it. And of course, I have the same question, right? Like I definitely am all into questioning things as well. I just know that there's like a limitation to this. You could take a series of different videos or see like different stages of a process and stuff. And then, you know, a lot is gained by actually doing this yourself. And then you could like just see it for yourself beyond what's just the only thing that's published in this paper, or whatever.
That's all junk.
Yeah. Sorry.
Well,
So
it's
if you do it,
that it's that the
So
electron microscope pictures that have been taken are of damaged cells, waste cells. There's not a virus that's isolated. The thing that's supposed to be causing this malformation to the cell. It's a picture of a damaged cell, and that's it.
it's not just a malformation of a cell, like it's an
Guys,
entire.
wait a second. This is Taylor's show. All right. I think it's a
Yeah.
Q&A, and I think that we should be interacting with her back and forth. So I
it's not a
think Taylor
damn.
should go ahead.
Well, it
I was
kind
just
of
gonna
is.
say
So she was
I've never
asked to demonstrate these
viruses.
things, and she hasn't.
Yeah, did you see my pictures of DNA and all that? Did you miss that?
Yeah, pictures. Yeah, great.
Well, what do you want? I mean,
Well, there's
I've never
video. I mean,
seen a
there's
virus.
video there too.
Pictures,
Yeah,
really? So if I
yeah.
provide a
I've
picture of
never,
Bigfoot, that's evidence, right?
I've never, like put any viruses in here. I'm talking about life. Viruses are non-living, so I don't study that virology. I study living cells. So I'm not going to sit here and defend something I've never personally seen, but I can defend
Where's a
DNA because
living cell?
we're made of them. You can look at them under a microscope.
Can you show me one?
Why are
Yeah.
Just
you so
one.
angry?
Yeah, I can show you. I don't know, but it's okay.
Why
Okay,
does
show
not
me some
believing in
shitty photo.
DNA
You already know.
No,
make
here's
one
some
an atheist,
videos.
though?
Okay, so I put some pictures of cells that
just
I've
look at the
taken
mirror,
here.
man.
Did you see this? This is one I've taken in pond water actually. Bacilleria paxillifera. And then here's another cell. It's a protist. You can
Wait,
see
yeah, but I
all.
was asking for a cell that we're made of, not some cell in a water.
The next one, these are human cells right here. These are neurons. This is a neutrophil. This is a white blood cell capturing a bacteria.
I'm not
And then
looking at it because I've seen all this stuff before and I know it's all junk. You cannot provide
you
an
won't
image of a
even look at what's on
cell
my
that a
screen?
human has. Again,
Yeah,
like I
these are
said,
three right here.
it's like listening to a globalist show me things and
This
then
is
them try
three
to tell me
cells.
that this is the curve of the earth.
Did you see? Well, then open my screen. You'll see 3
I
human
don't have
cells.
to. Again, I've already seen all this stuff.
Videos of
Whatever
them.
you're trying to present, there are no human cells. There's no images of these
Yeah,
things because they don't
there's videos
exist.
of human cells
I'll
right here
wait.
if you'd open
No,
it.
you're just like a globalist.
Okay, then open it. You're acting like them because you won't even look at your screen. And I'm telling you, I have videos of 3 cells on the screen right here.
I mean, if someone else wants to believe her, that's great.
Dude, you can get a microscope and look at it yourself. Like, you don't have to
Yeah,
believe
I'm
me.
sure. My
I
own
am.
cells. You're right.
You sure
I
can.
can just cut off a piece of my finger and then I can see individual cells.
You sure
Wrong.
can.
Doesn't
No,
work
that's
that
not
way.
wrong. No, yes, it does.
See,
You can
you
see.
think that because you're educated, you know more, but you don't.
I'm telling you, have you tried it?
You literally don't
Try
know
it
what I've
out.
done. You literally don't know
Try,
what I've
that's
done.
what I'm
And I
asking.
really don't
Have
have to
you
explain
tried it?
anything to you, but you're making claims that you can't back up.
I have seen my own cells under a microscope. I'm just asking you
No, you
if you've
haven't.
done it because I would think that you would change your opinion if you actually bought a microscope and tried it yourself.
No, you've never seen your own cells because they don't
How do you
exist.
know that?
That's just
I
stupid.
sure have. I've
Are
looked at
you
my
claiming that
cells in my
human cells
microscope. I
don't
stabbed
exist?
my
What are
teeth,
you going
looked at
on
my
about,
cells.
Lottie?
I'm
I
sorry.
mean,
Did he
show
say
me
there's
one.
no cells?
Yeah. I don't know.
This is wild.
You can get a microscope and look at them yourself. You can look at pond water. You can look at your cells.
Again,
Whatever
like
you,
I said, you have no idea what I've already done. You're
then
assuming
you
that you are
are
smarter and know more
right.
than I do,
I'm
but
not
you're
Just a
assuming.
presenting
reality denier.
cartoons.
I'm not assuming that. That's why I'm asking you if you've done it yourself, and I think that you could do it yourself.
Oh my god.
Right, and there's zero evidence for DNA or human cells.
There's no evidence for human cells and DNA. That's crazy. I have
Yeah, I mean,
videos.
I'll wait. I've been waiting for a while.
I have them on
soon
your
we're gonna
screen.
hear-
If you click on it, you see.
Pretty soon we're going to hear pictures from space of the Earth are fake too.
Hey, I'm
Do
just
squids have
curious.
cells?
I'm just curious, guys. Like, who else feels this way? I'm really curious.
Wait, are we going to talk about our feelings now? What is this, a woman's party?
Mm-hmm. Yeah.
See, that
Did you see
is
the stuff I posted in chat, Riggs? That's what it
Hey,
comes down
guys,
to. When
how
you're
do we
asking
feel
people
about
for
things
proof
that
of
other
DNA,
things are
those are
saying?
Human cells.
those are the
I do
images
believe
that
men
you're
have
given.
an
Work.
implicit function men are not men unless they can recognize through their feel of that situation a correct determination of the path they should proceed in. So yes, a feel that is telling you something, a sense, is definitely worth investigating at some point, not just ignoring into eternity.